Welcome to the Long COVID Coalition. My name is Leo Galland and I'm Head of Product Development at Medicinals Biotech. Together with Dr. Valentina Viduto from the Long COVID Foundation and Dr. Philip McMillan from Bayer Health, we are bringing the leading global experts on long COVID to the table in order to get a deeper understanding of the various multisystemic conditions at play. How COVID attacks the brain. Preview to the Long COVID Coalition Conference on Neuro COVID on June 25th, 7 p.m. UK time. The following slides are showing documentation and possible health consequences resulting from SARS-CoV-2 infections and exposure to SARS-CoV-2 spike proteins. We concentrated the selection of publications on so-called neuro-COVID documentations as to raise the awareness of possible pathways and which therapeutic interventions can be recommended. The flu pandemic from 1918 caused more than 1 million cases of viral Parkinsonism. All this sounds quite alarming, so let us see the evidence, let us listen to the experts, see the data, see the science behind it, and what can be done to stop disease progression and which solutions are available.
I'm Dr. Leo Galland, and I'll be speaking at the 4th COVID Coalition Congress. I look forward to seeing you then.
Hello, everyone. Hi, my name is Dr. Kamal Dua, and I am from the discipline of Pharmacy, Graduate School of Health, University of Technology, Sydney, Australia.
Hi, my name is Dr. Valentina Viduto. I am founder of Long COVID Foundation. Look forward to meeting you there. Bye.
Hello, my name is Rachel Jessey and I'm a registered naturopathic nutritionist, and I'm really looking forward to presenting here at the 4th Long COVID Coalition.
Hi, I'm Dr. Shankara Chetty, a frontline worker and natural science biologist. I'm glad to be part of the 4th Long COVID Coalition.
Hello, everybody. My name is Stephanie Seneff. I'm a senior research scientist at MIT. I'm very much looking forward to having the opportunity to speak at the 4th Long COVID Conference.
Hello, my name is Leo Galland and I'm Head of R&D at Medicinals India and a founding member of the Long COVID Coalition.
Hi, my name is Abdul Manan Beg. I'm from Khan University at Karachi, Pakistan. Today I'm going to talk along with my peers on this topic at the Long Coalition Congress. Thank you very much.
Hello, everyone. Let's start again. Tremendous honor to have everyone here today. And we have a big panel of people, speakers, panelists to share with you the latest thinking in long COVID focused on neurology. And without further ado, what I'll do is I'll bring in all of our speakers and panelists who will do a quick hello to everyone. Thank you all very much for joining us from different parts of the world. And we have a special thank you to Dr. Kamal Dua, who has come all the way from Australia to be able to share with us. And he is woken up in the middle of the night. Tremendous. Thank you very much, Kamal, for being here with us. So before we start anything else, we will just want to hear a very quick word from Dr. Valentina Veduto, who is one of the founding members of the Long COVID Coalition. So we'll just give her a few minutes just to share a few thoughts before we introduce our moderator by myself. So you can go ahead, Valentina.
Thank you very much. So, distinguished speakers, panelists, and viewers, it is my great honor to participate in the 4th Long COVID Coalition Congress on Neuro COVID. We are very glad to meet again and, of course, share most relevant scientific findings surrounding this novel disease. At the Long COVID Foundation, our vision is to improve health and well-being of Long COVID sufferers by contributing to the Long COVID disease's understanding and treatment options. And to do this, we work with organizations and individuals of medical professions, scientists, and researchers, and Long COVID interest groups to promote awareness of long-standing symptoms, health, and rehabilitation challenges. Long COVID is a new phenomenon, a new disease that, if not treated, can lead to long-term issues. As a founder, I see the huge limitation in the healthcare system, and which is still catching up with the science. Therefore, at the foundation, we provide people with appropriate information and knowledge to have a healthier and more sustainable long COVID recovery. Long COVID Coalition is established to bridge the gaps in knowledge and identify the solutions which address these problems. And jointly with medics and independent scientists and researchers, we bring the most up-to-date research on long COVID. And today's Congress is just another global challenge to solve neuro COVID. And I'm delighted to participate today, and I look forward to help you understand more. Thank you.
Thank you very much, Valentina. And straight away, what I'd like to do is introduce our moderator for today. And it's Dr. Kamal Dua, who's all the way in Australia. And as I said, we really appreciate that he has been able to come with us. And this just shows how important this international flavor that we have on neuro-COVID is. So Dr. Kamal Dua is an award-winning pharmaceutics and drug delivery researcher who has deep roots in teaching, research, and industry spanning 3 nations and 2 continents. He holds dual expertise in pharmaceutical sciences and immunology and microbiology, and he also completed 4 professional diplomas in the areas of clinical research, product development, and drug regulation. Kamal's academic research has contributed significantly to the field of managing chronic inflammatory diseases, especially airways diseases, using novel nanocarriers to deliver potential therapeutic molecules, including phytoconstituents. His research identifying potential key targets, regulating the pathophysiology of the disease, and formulating them into effective drug delivery systems that could be translated into clinical settings. So tremendous to have you with us, Kamal, and I'll allow you to introduce our first speaker, who will be Dr. Leo Galland. Thank you very much for joining us, Kamal.
Thank you, Philip. That's a pleasure. Thank you so much. Hello, welcome everyone to the 4th Long COVID Coalition Congress on Neuro-COVID. Valentina has nicely presented already what's the relevance and importance of this Coalition Congress. And without any delay, I would like to introduce our first speaker, Dr. Leo Galland. Dr. Leo Galland is a pioneer in studying the impact of intestinal microbes, the gut microbiome, and intestinal permeability, which is called leaky gut, on the health and disease. He has received international recognition for developing innovative nutritional therapies to treat autoimmune, inflammatory, allergic, infectious, and even gastrointestinal disorders. He has described his work in numerous scientific articles and textbook chapters. He's a graduate from Harvard University and New York University School of Medicine. Dr. Galland is board certified in internal medicine. He's listed in leading physicians of the world and America's top doctors. In 2017, Dr. Galland was awarded the Albert Nelson Marcus Lifetime Achievement Award by Marcus. Who's Who, Dr. Galland, Who's Who. Dr. Galland is regularly chosen by New York Magazine as one of the best doctors in New York. So without any delay, I would like to invite now Dr. Galland to please share his screen and deliver his talk. Thank you. Dr. Galland, you are on mute. Could you please just unmute yourself? Thank you.
Thanks. Got it. In the past 10 days, there have been 4 major papers that have appeared that I've had to add to this presentation because this is such a dynamic area. And I expect that this is really just a work in progress, and it is based on my reading of the literature and the practical steps that I take with patients who have long COVID. There is a roadmap that I attempt to follow to reestablish a healthy gut-brain axis after COVID-19. It's well known that there are disturbances in the gut microbiome associated with COVID-19. Some may predispose to severity of COVID, some predisposed to long COVID, and I'll highlight those, and others are probably the result of having COVID. And I think those are becoming increasingly important. The steps that we will follow are to stop viral proliferation in the gut, reduce epithelial cell inflammation that's in the gut, reverse the gut bacterial dysbiosis that is created by COVID-19, restore the transport and metabolism of tryptophan, which is impaired in people with COVID-19, restore the mucus layer in the GI tract and tight junction integrity to assure a return to normal intestinal permeability, detoxify the intestines, and then I want to create a microbiome that will support T memory cell function, which is essential not only to recovery from neuropathic, but also to protection against the effects of future infection. Now, there's abundant evidence that persisting viral infection contributes to long COVID, or PASC, as it's called in the US in scientific circles. I'm not going to review all of these studies. The most recent was a study from Harvard which found circulating spike protein in the blood of people, in the plasma, not even in cells, of people with COVID-19 up to 12 months after they'd first been infected. And these researchers said there has to be a reservoir of chronic viral infection that is present and that is creating this and then driving, presumably driving long COVID. And the immune response of people with neuropathic in particular, this was studied at Northwestern University and is referenced here, shows an impairment of T effector memory cells of a type that would be seen with chronic antigenic stimulation. So steps to prevent viral proliferation in the gut. There are drugs that might be used. I mostly are using natural products, and flavonoids figure very strongly into that pharmaceutical repertoire, especially those containing quercetin, curcumin, bicalin from Scutellaria bicalensis, glycyrrhizin from licorice. They have effects of protease inhibition, and they also destroy bacteriophages. And as Valentina will be explaining later, and there was data presented on this at the last conference, there is very strong evidence that the virus, the SARS-CoV-2 virus, not only infects human cells, but can infect bacteria and act as a bacteriophage. Based upon that data and the work of Dr. Carlo Brogna and his colleagues at Cardiomed in Italy, I am now exploring the role of antibiotics along with these antiviral herbs, in particular amoxicillin, which in his studies actually proved to be the most effective. I'm also exploring the use of oregano oil, because oregano oil has both antibacterial and antibacteriophage activity. And in a review of dietary factors that could impact the activity of bacteriophages, polyphenols, especially the bioflavonoids, were identified, and oregano was found to suppress multiple types of prophages, preventing them from incorporating into the bacterial genome. And what bacteriophages do is they are viruses that enter into bacteria as become prophages, then become incorporated into the genome, activated, and they may kill or lyse the bacteria or change the metabolic activity of the bacteria. Of note, the natural sweetener stevia was shown to promote the activity of multiple species of bacteriophages, increasing their impact on bacterial hosts. So since we don't know what stevia does in this particular setting, I would use caution with stevia in patients who've had COVID-19. Now, there also is a probiotic that has antiviral activity. It is Bacillus subtilis B7092. It is a particular strain. If you can find this, when I have access to it, I use it. It was developed in the Soviet Union about 40 to 50 years ago for the purpose of delivering human alpha interferon through a plasmid into the GI tract. And SARS-CoV-2 virus is very sensitive to killing by alpha interferon. It's available in a few countries. I've mentioned the names. It was actually developed in Ukraine initially. So I will use, in the initial stages, I will use these antiviral herbal products that are rich in flavonoids, along with antibiotics, and then follow that with Bacillus subtilis B7092. The next step is to reduce epithelial cell inflammation. The idea that you can just go and correct the dysbiosis of an inflamed intestine with probiotics or prebiotics, I think is incorrect, because inflammation itself alters the milieu. Inflammation increases the synthesis of nitric oxide, which results in an increase in the nitrate concentration of the inflamed tissue. Now, a high nitrate environment, which is what's created, enhances the growth of many bacterial pathogens, but it inhibits the growth of many healthy anti-inflammatory natural bacteria. We see the same thing in streams and in water when high nitrate detergents alter the nitrate concentration, or when high nitrate fertilizers run off into the water. This happens in the gut. And so this is a vicious cycle. The high nitrate environment invites the growth of pathogenic bacteria, which then generate more inflammation and maintain this nitrate cycle. Now, there are a number of, so basically the foundation for reversing gut bacterial dysbiosis requires controlling intestinal inflammation. There are a number of natural products that I use for this purpose. Quercetin, mastic gum from the Mediterranean food Pistachio lentiscus, curcumin from turmeric, omega-3 fats, magnolia bark, which is used a lot in Chinese medicine, and bovine serum immunoglobulins have all been shown in controlled studies to reverse intestinal inflammation, and I will choose from among those as part of the treatment. Because some of these also have antiviral effects, they do double duty. Quercetin has especially been studied for its antiviral effects and its anti-inflammatory effects in the gut, and there's several studies to that effect. Now, improving the gut microbiome. There is one study which is consistent with the results of many others, which showed that the gut microbiome at disease onset predicts the development of PASC. Previous studies had looked at people who had been hospitalized with COVID and looked at them after hospitalization and found very similar features to these features which were found to predispose to the development of long COVID. People who did not show these features had a much better outcome and recovered fully from COVID-19. First of all, there is a shift in bacterial concentration, and I've highlighted what I think are the three most important findings. There is a keystone bacterial species called Faecalibacterium prausnitzii, which is a major producer of a short-chain fatty acid called butyrate that has important anti-inflammatory effects in the GI tract and in the body as a whole. F. prausnitzii is diminished. There are higher levels of certain other bacteria that are associated with inflammation. In particular, a species called Ruminococcus navis. Now, you may not have heard of these. These are not available as probiotics, but they are very important, and the balance between them is very important in gut health. Bacteria that produce butyrate, their activity was inversely associated with the risk of developing long COVID. And F. prausnitzii is a major butyrate producer. So butyrate protects against long COVID. Loss of butyrate predisposes to long COVID. When we look at the physiology of butyrate, it becomes pretty clear that this is not just a coincidence. So step 3 is to try and restore the depleted bacteria and restrain the inflammatory bacteria. Once again, in a general way, bioflavonoids play a major role in doing that. Flavonoids I view as curators of the gut microbiome, and they encourage the growth of and activity of certain species and inhibit certain other species. And at the same time, we need these flavonoid-dependent bacteria because they make the flavonoids more available to us. So butyrate is what is called a postbiotic. It is produced by bacteria in the gut. It's not produced by human cells. It has nutritional effects in the gut. About 80% of the energy that maintains the integrity of the lining of the large intestine comes from butyrate. The mitochondria in the large intestine depend on butyrate for their activity. It is produced by gut bacteria that ferment certain kinds of fibers and complex carbohydrates. Outside the GI tract, butyrate has some important effects. It has effects in the brain. This is a volatile short-chain fatty acid. It passes very readily through membranes. What's produced in the GI tract goes into the body. What circulates in the blood goes readily into the brain. One of the systemic effects of butyrate, it is an inhibitor of an enzyme called histone deacetylase. Histone deacetylase inhibits certain genes, and it has been shown that in the brain, the presence of butyrate activates some quiescent genes, in particular, a gene that's involved in producing a protein called brain-derived neurotrophic factor, BDNF, which is really important for recovery of neurons from damage and injury. It also activates various complexes on cells called G protein coupled receptors. Some of them are only activated by butyrate, and they have important effects in regulating immune responses and inflammation. So the steps to be taken toward reversing gut bacterial dysbiosis among people with long COVID. Number one, from a dietary perspective, the science would suggest a high fiber, polyphenol rich diet that increases butyrate secretion production. It increases the growth of Faecalibacterium prausnitzii. I have some colleagues who prefer other types of diets. In looking at the science of what will correct the gut dysbiosis that follows COVID-19, a high fiber, plant-based, high polyphenol diet really is the one that meets the research criteria. And an adherence to a Mediterranean type diet was also shown to elevate fecal levels of Faecalibacterium prausnitzii. There are prebiotics that can be used, oligosaccharides, fructooligosaccharides, galacto-oligosaccharides that increase the growth of F. prausnitzii. Resveratrol is something that I've used for a different effect. There's a species of strongly inflammatory bacteria called Enterococcus faecalis that's associated with worse outcomes in people hospitalized with COVID-19. It is a major stimulus to the production of gamma interferon, which plays a role in the cytokine storm of acute COVID. Resveratrol inhibits the growth of E. faecalis, and also has effects in modulating the ability of bacteria to produce superantigens in the respiratory tract. There are two species of probiotics, strains that is, of probiotics, that have been shown to enhance the growth of F. prausnitzii in controlled human clinical trials. One of them is Bifidobacterium longum BB536. This was studied in children and adults with allergies, and at the same time that it had anti-inflammatory effects, it increased the growth of F. prausnitzii. And then there's a soil-derived organism called Bacillus coagulans, a particular strain, GB130, which was studied in elderly people and found to decrease inflammatory markers like C-reactive protein and increase the growth of F. prausnitzii. So I will often use those following the treatment with Bacillus subtilis. Now, this is a study that just came out on Ruminococcus navis. Now you'll remember that higher levels of Ruminococcus navis was associated with an increased risk of long COVID. It turns out that Ruminococci, the whole genus that includes this, produce substances that may be toxic to the brain. In particular, there's a substance called isoamylamine, which when studied in rats was shown to quicken the pace of senescent cognitive decline. Stephanie will be talking about senescence later, and I think that the senescence, accelerated senescence in the brain, is part of the problem we're dealing with. Ruminococcus or Ruminococci and their metabolites may play a role there. Ruminococcus navis also converts tryptophan, an essential amino acid, to tryptamine, an amine that promotes migraine headaches and migraine phenomena. And migraine type phenomena play an important role in the physiology of neuropathic. So I'd like to move on to amino acids as they interact with the gut microbiome. We could spend a whole day talking about that. I'm just going to mention two of them. Tryptophan is converted to serotonin and to melatonin. Phenylalanine, another essential amino acid, is converted to tyrosine, which is an amino acid that is then deaminated to dopamine, decarboxylated to dopamine and norepinephrine, which are neurotransmitters. There are bacteria that produce these neurotransmitters, and there are bacteria that respond to their presence. What's especially important for ACE2, and I think Rachel will be talking about this more for COVID-19, is that ACE2, the cellular receptor for COVID-19, which is destroyed by interaction with the spike protein, has a special role in the gut. It is the chaperone that aids in the absorption of large neutral amino acids that include tryptophan and phenylalanine. And in fact, when tryptophan, when there is impairment of ACE2 activity in the gut, tryptophan is the most effective. Okay, so one of the goals is to restore tryptophan serotonin balance. And we start by enhancing ACE2 activity. There are a number of natural products that will do that: vitamin D, curcumin, resveratrol, variety of antioxidants, omega-3 fats. Fortunately, these have played other roles in antiviral roles and anti-inflammatory roles. Quercetin has complicated effects. It inhibits the breakdown, the oxidation of serotonin, but there was a study indicating it may reduce serotonin synthesis. So typically there's a point after about 2 or 3 weeks where I will pull serotonin out of the treatment to see how the person does without, to pull quercetin out of the treatment to see how the person does without quercetin. Tryptophan supplementation might be beneficial. There's an animal study that suggests that. I have not really been utilizing tryptophan in my treatment of patients with long COVID, but after reviewing this research, I will be looking into it. And there is a tryptophan metabolite called indole-3 propionic acid, which is produced by gut bacteria, in particular bacteria of the Clostridium species. That's a very complex species with some dangerous organisms and some very important ones. Clostridium sporogenes is the one that's been identified as the major producer of this. This has anti-inflammatory effects. It has neuro-restorative effects. And so there may be a role for tryptophan and balancing of gut flora to increase production of IPA for healing the nervous system. Restoring the mucus layer and healing tight junctions are both important for restoring normal permeability in the intestinal tract after COVID-19. Many of the steps that we've already taken will do that. Bicalin, quercetin, the probiotic changes will all help with that. Finally, there's the question of, is there still toxicity in the infection, in the intestines? And is there something that needs to be altered? And this may even wind up being an earlier step in the process. One of the studies that was done, a couple of studies done with people hospitalized with COVID-19 showed fungal overgrowth in their stool persisting after discharge. And this fungal overgrowth was associated with fungal species that produce mycotoxins, as well as some pathogenic yeasts like Candida auris and Candida albicans. Now, there's a recent study in the US that found fungal cell wall beta-glucans circulating in the blood of people with long COVID. And so there are a number of steps that should be taken to remove these fungi and their toxins and detoxify the intestines. Activated charcoal is one of them. I've been exploring the use of antifungal herbs as well. And then finally, enhancing the function of T effector memory cells, which are depleted in people with neuropathic. This may not occur across the full spectrum of patients with long COVID, but in a study from Northwestern University in Chicago, it was very clear that the function of T effector memory cells was one thing that all people with neuropathic had in common. Now, the gut microbiome has an important impact on the development of T effector memory cells. they butyrate, and fats and fatty acids in general enhance them. Fermented foods, lactobacilli in particular, enhance their development. And so in addition to a high fiber, adequate fat diet, fermented foods like sauerkraut and kimchi are warranted. They are very dependent upon mitochondrial function. So at some point, we really need to have a symposium devoted to mitochondria in long COVID, because they show up everywhere. And this last slide is about Lactobacillus plantarum, which is found in fermented foods. There are also supplements. It grows on plants. There have been a couple of controlled studies, not in long COVID, but in other conditions in which Lactobacillus plantarum was really shown to impact immune function and recovery from infection in a very positive way. Thank you for your attention.
Thank you.
Thank you so much, Dr. Galland. I should appreciate the slides and information that you've managed to combine into these slides. And I know that you've looked into tens papers during this short period of time. And I should appreciate that you've designed such a beautiful presentation and the pack of knowledge for us to understand where basically neurological symptoms start. So gut health is really important. And as a representative for people who struggle to understand simple things like what type of probiotic they need to get, and I know you focused on Tundrex as a supplement to address antiviral properties that happen in long COVID. But if people can't get this particular type of probiotic, what is your view on going for a combination of different species, and the more species you get as a probiotic in a single solution is better, or people still need to focus on the key species, that keystone species you've mentioned?
Well, the keystone species that we want to build up, F. prausnitzii, which supports the growth of many other species, especially bifidobacteria, you can't get that as a supplement. So you have to build it up indirectly through diet, prebiotics. And then I showed slides on two probiotics that were shown to increase its growth. I don't think that the use of the Bacillus subtilis is essential. Also, it's a very short-term use. It's 2 or 3 weeks, no longer. We just want to get, what my goal is to try and get the spike protein out of the GI tract. Make sure that there's no replicating virus left. I can't guarantee that we're doing that, but that certainly is an immediate early goal. Then I want to try and build up a healthy gut microbiome, reduce the growth of ruminococcus, and a couple of things on that slide, by the way. Malvidin, a bioflavonoid found in berries, especially blueberries, inhibits the growth of ruminococcus navis. And again, a high fiber, plant-based, polyphenol-rich diet inhibits the growth of ruminococcus at the same time that it's building up Faecalibacterium prausnitzii. So I try to accomplish that mostly through diet, select probiotics, and Carlo, by the way, uses lactobacillus in his protocol. And so at some point, I will move over to lactobacillus species, usually the plantarum, as part of the attempt to enhance T memory cell activity.
I see. So overall, how to make sure you have quite good diversity of good bacteria, rather than you increase the rate of not good bacteria, which is bad and works against the good bacteria. So all these combinations with many species of bacteria, so it's not necessarily working good as far as I understand.
You know, I mean, there are 1,000 species of bacteria. So if you take a probiotic with 12 strains, most of which are going to be lactobacilli and bifidobacteria, which are really, the lactobacilli in particular, are really minor components of the normal gut microbiome. That doesn't mean they're not important, because number isn't the same as importance.
Yes.
But just taking more species rather than fewer, there's really no evidence that multi-species probiotics perform better.
Thank you. Thank you.
So,
oh, and I just want to add that this is a very individual thing. You need to work, you need to know your body. You need to pay attention to what's happening. I mean, I have patients who are taking multi-strain probiotics and they say, I really like this. This is the one that has helped me the most. I say, fine, stick with it. I mean, if you're getting that response to it, you're getting feedback from your body, pay attention to it.
Sure.
Dr. Galland, I got a couple of just questions for you. So when you talk about high fiber polyphenol diets, I think quite a number of abundant food contains, high fiber, those polyphenols. And there are a lot of nutritional supplements also being easily available in the market. So what do you suggest people should take? Those supplements will do the same as what if they take those natural fruits or whatever the sources they have in terms of the highest polyphenol contents and fiber?
Oh, okay, so in general, as a general principle, my belief is if you can do it with food, do it with food. However, a lot of people following COVID-19 will have GI symptoms.
Right.
And they may or may not be able to follow the kind of diet that I'm describing. So again, this is something you have to work out for yourself. I mean, the simple approach, especially for your people in Europe and North America, is to look to the so-called Mediterranean diet as a starting point. And there's just been so much written about it. In other parts of the world, look to traditional diets in the countries that you live in. I mean, the traditional Indian diet, the traditional Chinese diet, because traditional diets tend to meet those criteria. They tend to be plant-based and high in fiber and high in polyphenols.
Right.
And that's not an accident.
Sure. So Dr. Galland, just continuing on that question. So when we say, you get from your own diet, is there a particular concentration you're looking for all those polyphenols or high fiber diets from that? Is that what you're talking about, a specific concentration?
The total amount of polyphenols from diet, I mean, I don't think anybody plans their meals this way, but should be in the range of 4,000 milligrams a day.
Right.
And it is, for most people, it's hard to overdose on those. But there are some people who have certain genetic susceptibilities or genetic problems. They're not going to tolerate that because one of the characteristics of bioflavonoids is they inhibit a lot of enzymes. And if you are genetically predisposed to, because the certain enzymes are weak, like the enzymes that methylate neurotransmitters, your tolerance for bioflavonoids may be much lower than someone else's.
Sure. So, Philip, you on mute. So just a last question. Do you think that the antibiotic can be mixed with polyphenols, like amoxicillin?
Yes. Now there are yes, I think amoxicillin can be. There are certain antibiotics that in which the polyphenols might actually interfere with the receptor binding, but not like the quinolone antibiotics, but not in this setting.
Right. Thank you. Thanks, Dr. Galland.
Yeah,
Thank
I
you
just
so
wanted to ask a quick question from the audience here, Leo. This is from Rick. How long does it take for the virus to clear the GI tract in long COVID?
Without specific treatment, it may last at least a year. And there was a study that was done in people who were getting routine colonoscopies because of inflammatory bowel disease after having had COVID-19. And the people who had long COVID, but not the people who had fully recovered or the people who never had COVID, actually had spike protein in the cells. Not only spike proteins, there were nucleocapsid proteins in the cells lining the large intestine 12 months after having had COVID. And there was a hyperstimulation of certain types of immune cells in those specimens as well. It's actually certain types of T cells. That's a very complex process that we're going to need to really try and tease apart.
Wonderful. I'm going to ask us to move on to our next speaker now. So I'm going to shift over to Dr. Kamal, and I'm certain we'll have more questions later on, Leo.
Thank you. Thanks, Dr. Galland, again. It was really a great presentation. So let's move to our next speaker, Rachel Jessey. Rachel Jessey is a registered naturopathic nutritionist and founder of BeNourished, which is a UK-based nutrition and lifestyle consultancy agency, and that is leading the way in personalized nutrition for supporting resistance, resilience, and recovery for chronic illnesses. Rachel have enjoyed 12 years of clinical experience, and she has gained extensive knowledge in the nutrition and integrative healthcare space and works with clients both nationally and internationally. Rachel has been researching and working exclusively on long COVID for the past 2 years and helps her patients to identify the root cause of disease, processes, and develops personalized nutrition and lifestyle protocols to support recovery. So let's welcome Rachel Jessey. You can share your screen, Rachel. Thank you.
Thank you so much for that introduction, and good evening, everyone. I'm really happy to be presenting to you all today. Dr. Leo Galland, thank you so much for that excellent presentation, really informative information. So today I'm going to be talking through the kineranine pathway. So we're going to start with talking about tryptophan metabolism, and then I'm going to take you on a deep dive down the kineranine pathway and try and explain that in the best way I possibly can. And then we're going to look at a little molecule called NAD+ and why this is important. And then we're going to look at how the kineranine pathway is affected in COVID-19, so how neurological function is affected in COVID-19 and long COVID. And then I'm just going to talk you through some of the interventions that I have been using in clinics. So they're primarily dietary and lifestyle related. So I want to note that I'm going to be referring to this pathway in the context of neurological function alone. So before we dive into the pathway, let's have a quick look at tryptophan. So tryptophan is an essential amino acid. This means that our body can't make it, so we need to get it from our food, and around 1 to 10% is going down the serotonin pathway. A portion of that gets metabolized into indoles, and then the rest, around 95%, depending on what publication you're reading, enters the kineranine pathway. And then we've got this quite scary diagram here. So this is the kineranine pathway. So I'm going to try my best to break this down as simply as I possibly can. So this diagram here is basically showing you the pathway in detail. And as you can see, there are several conversions and metabolites that are produced along each of these stages. And each of the metabolites are going to exert a specific function on the body. And when this pathway works well, it's working beautifully because it's controlling inflammation, it's supporting our immune function, it plays a massive role in neurological function and also energy production. So a couple of things that I want to note along this pathway is that the metabolism of tryptophan down into kynurenine depends on the IDO and the TDO enzymes. And kynurenine itself plays a really important role as what we call an immune checkpoint molecule. So it's regulating various immune responses via a feedback mechanism related to the aryl hydrocarbon receptors. Now, I'm not going to talk about that, but this is basically to note that this top portion of this kynurenine pathway is involved in immune function. And then kynurenine can be converted into differing metabolites via 3 specific enzyme pathways. So this enzyme pathway here, so kynurenine into kynurenic acid, takes place in the presence of the CATS enzymes. And kynurenic acid is an N-methyl D-aspartate antagonist. So what does that mean? Now, I'm going to refer to N-methyl D-aspartate as NMDA. And basically, what NMDA does is it mimics the action of glutamate at the NMDA receptors. So it exerts an excitotoxic effect. So basically what kynurenic acid does is it blocks this receptor. So it exerts an anti-excitotoxic and anti-convulsant effect at the receptor. So kynurenic acid is generally considered to be neuroprotective. And then you have the KMO enzymes and the kynurenase enzymes that basically push kynurenine down this pathway to ultimately end up as 3-hydroxy and anthranilic acid. Now, this section of the pathway generates quite a lot of free radicals and reactive oxygen species, but in the presence of that, there's also going to be an activation of the antioxidant system. So, you know, this is just another example in this pathway of balance. And then when we get to 3-hydroxyanthranilic acid, this can be converted down the pathway to quinolinic acid or picolinic acid. Now, quinolinic acid is really important because this is an NMDA agonist. So it's got the opposing function to kynurenic acid, and it's also referred to as an excitotoxin. And picolinic acid is thought to be a neuroprotector. Now, the pot of gold at the end of the kynurenine pathway, this is what I refer to it as, is this molecule here, NAD+. And then you can see that all of these nutrients are involved in the conversion steps of this pathway down to NAD+. So we've got vitamin B6, B2, magnesium, and B3, which we know as niacin. And then this second diagram, which I'm not going to go into great detail in, but I just wanted to highlight that the kynurenine pathway also relies on the niacin-dependent Priest-Handler pathway, which begins with nicotinic acid or niacin, B3, however you want to call it. And it's important in the process of generating NAD. So basically, niacin is going to be pushed down this pathway. It's going to meet with quinolinic acid, and it's going to produce NAD+. And also here, up here, we've got the CD38 pathway, which is a major consumer of NAD. And that feeds niacin-like metabolites down into the NAD salvage pathway. So really, this whole mechanism is responsible for recycling and generating NAD+. So why is NAD so important? So It's got a lot of functions within the body. So one, it's supporting a healthy mitochondrial function. So it's involved in glycolysis, it is involved in the citric acid cycle, it's involved in oxidative phosphorylation and the generation of ATP. So you can break that down into it's important for energy production. And it also helps to balance T cell metabolism. So it's stabilizing our macrophage phenotype. So we have a tight balance between the inflammatory macrophages, known as the M1 macrophages, and the M2 macrophages, which are our resolving macrophages. So you can see that NAD+, if it goes out of balance, then you're going to get an imbalance within the immune system. And then we have protein molecules called sirtuins and poly-ADP ribose polymerases, which I just refer to as the SIRTs and the PARPs. Now, these are major consumers of NAD+, and what these protein molecules do is that they protect our genes, so they're having a direct effect on our epigenetic expression. And then the other cool thing about NAD+ is it regulates circadian expression of important metabolic genes. It's sensing things like nutrient availability, insulin, it helps regulate sleep and wake cycle, and it's also heavily involved in reproduction as well. So you can see why it's really important that we make this molecule from a properly functioning kynurenine pathway. So how does this all translate into COVID-19? Well, there have been a couple of human studies looking at the effect of COVID-19 on the kynurenine pathway. And we'll start off with this, this first study that was done in 2020, which found that in response to increases in interleukin-6 during COVID-19 infection, this was corresponding with a shift in tryptophan metabolism towards the kynurenine pathway and away from serotonin production. And it's also generally well accepted that IL-6 and other inflammatory cytokines are actually potent inducers of these IDO enzymes. And if you remember, the IDO enzymes are responsible for pushing tryptophan down into the kynurenine pathway. So these are upregulated in response to cytokines. And next we have another study. This is more recent. This was done in 2022 that found that a lot of important markers within the kynurenine pathway were significantly different in COVID-19 patients when compared to control. And according to the results of this study, kynurenine metabolites play a really important role in the pathophysiology of COVID-19, and there seems to be an increased ratio between kynurenine and tryptophan. And interestingly, in this study, they found that picolinic acid was also raised. And if you remember, picolinic acid is neuroprotective. So this is suggesting that this pathway is trying really hard to gain some balance and control over excess inflammatory processes. So on the back of these studies, there's been quite a lot of research into the kynurenine pathway and how dysregulation of this pathway could be implicated in long COVID. So this first diagram here is showing you that in response to spike protein, you're going to get diminished ACE2. And this is going to inhibit major amino acid transporters, which are here. And these transporters are responsible for getting tryptophan and other amino acids across the gut wall and into the bloodstream. So you're looking at a potential amino acid malabsorption here within the gut. The other mechanism at play is that in the presence of persistent inflammation, so these inflammatory cytokines, there's going to be increased expression of the IDO enzymes, which further increases the conversion of tryptophan down into the kynurenine pathway. And these mechanisms are thought to persist in those experiencing the lasting neurological effects of COVID-19. So let's visualize this information in the context of the main functions of the kynurenine pathway. So we've just found out that in the presence of disrupted tryptophan absorption and upregulated IDO enzyme activity, we're going to see tryptophan pushed down into the kynurenine pathway at the potential expense of serotonin production. Now, when excess kynurenine is produced, it's preferentially directed down this enzyme pathway here, the KMO enzymes, as opposed to the CATS enzymes. So at this point in the pathway, there is a potential that there's going to be insufficient production of neuroprotective kynurenic acid for the upcoming storm that's going to be coming further down the pathway. And this is further exacerbated in a vitamin B6 deficiency because we need vitamin B6 to make these CATS enzymes function properly. And so as you see a push down into this pathway, you're going to start to see an excessive production of 3-hydroxykynurenine and 3-hydroxyanthranilic acid, which will exacerbate the production of reactive oxygen species. And you're going to start to see that overwhelming the antioxidant capabilities. And then a further push down into this pathway is going to result in mitochondrial membrane destabilization, disruption to the electron transport chain, and this is where fatigue is going to start coming in, brain fog, all of these symptoms. And then what will happen is you will go down into producing quinolinic acid or picolinic acid. Now, although some neuroprotective picolinic acid is going to be produced, these 3-HAO enzymes here are very, very efficient at pushing 3-hydroxyanthranilic acid into kynurenic acid. And on the opposite effect, this QPRT enzyme is a little bit slower at converting quinolinic acid to NAD+. So what you're going to end up with is a bottleneck here of quinolinic acid, and that's going to increase the NMDA receptor response. It's going to increase more reactive oxygen species, and it's going to produce excitotoxic and neurotoxic effects on the brain. So lastly, actually, I wanted to point out something extra along this pathway referring to the TDO enzymes. So these TDO enzymes are upregulated by glucocorticoids, in particular cortisol. And this pathway, I believe, is also relevant to long COVID, because with chronic health symptoms also comes fear and health anxiety, which I'm seeing a lot within my own patients. And all this is going to result in is kind of a positive feedback loop where, you know, with increased fear, increased health anxiety, it's going to drive up cortisol levels. This is going to upregulate TDO and drive more tryptophan down into the kynurenine pathway. And then if you've got more stress and more anxiety and more cortisol, you are going to deplete further the B vitamins and the magnesium needed for all of these conversions within this pathway, and particularly quinolinic acid to NAD+, which is so dependent on niacin, which is a B vitamin, and also magnesium. So in a nutshell, excess quinolinic acid is going to have neurotoxic effects in quite a major way. And the primary mechanism behind this is largely related with the overactivation of these NMDA receptors. So what's going to happen when these are overactivated is you're going to see an increase in calcium ions, and this is going to negatively impact on our antioxidant capability. So it's going to reduce superoxide dismutase and glutathione peroxidases, but also what you're going to see is an activation of inflammation through the COX and LOX pathways. And also to note is that quinolinic acid also directly interacts with free iron, and this joins together to form a kind of toxic complex that exacerbates oxidative stress and induces lipid peroxidation, increases peroxynitrite, which is toxic, and then you're going to end up with mitochondrial dysfunction, ATP exhaustion, and then you're going to get cytochrome C release, which is ultimately going to lead to the death of the cell. And eventually these toxic signals can be extended to reach other cells within the brain, and this is what starts the degenerative chain reaction. And so this quite impressive infographic here is kind of summarizing all of this. So when we tie it all together into how the brain is potentially being affected by the spike protein, you can see that once the immune system is activated, you're going to get a push in the or an increased release of cytokines and microglia activation, which is going to result in increased kineranine, increased quinolinic acid and glutamate, and this is going to deplete serotonin production, dopamine and norepinephrine, which kind of switches all of these mechanisms off. And so you're going to lead to altered neurotransmission. There's going to be neuronal damage and increased excitotoxicity, and there's going to be hypoxic injury, and all of these are contributing to these neuropsychiatric and also neurodegenerative symptoms that we are starting to see in long COVID. So after painting a relatively gloomy picture of the tryptophan kynurenine pathway in long COVID, there are many things that can be done to support this pathway. I want to mention that the information that Dr. Leo Galland has just presented on the use of antioxidants, flavonoids and polyphenols such as vitamin E, quercetin, EG, CG and curcumin not only support gut function, which is something that I'm looking at optimizing as well, but it also helps to mitigate the detrimental effects that can be seen in a dysfunctional kynurenine pathway. So supporting mitochondrial function, offsetting the detrimental effects of reactive oxygen species, improving the absorption of amino acids across the gut wall and helping to balance inflammation. So I won't touch too much on all of these because they've been covered in quite a lot of detail already, but there's some further logical aspects that I consider when supporting this specific pathway. We know that excess inflammation, excess stress, increased reactive oxygen species, poor gut health and nutritional insufficiencies are putting pressure on this pathway. So to me, it makes sense to address all these factors. And so my approach is very much about going back to basics and addressing these foundational aspects of nutrition and lifestyle. So on top of addressing the gut and antioxidant and polyphenol intake, I perform a dietary analysis. I will use testing to assess the kynurenine pathway, and I will look to address any of the nutritional deficiencies that I find, particularly those involved in this particular pathway. For instance, I'll be looking at protein, magnesium, B vitamins and iron, which all play a vital role in the downstream cascade of the conversion of kynurenine to NAD+. And so when I'm looking at optimizing dietary intake, from my perspective, this is highly personalized. So there is not one size fits all here. But what I'm finding is, or what I'm going to discuss are the general trends that I'm seeing in my clinic. So don't take this as specific advice. This is just kind of what I'm noticing. So what I'm finding is that people are better off on organic and glyphosate-free food. Some patients are doing really well on low histamine diets. Others are doing well on low oxalate diets. And this is because some of the bacteria that are responsible for degrading oxalate in the gut can be diminished, in which case I'll be taking oxalates out until we support gut function. And from a protein perspective, I'm using anywhere between 0.8 and 2 grams of protein per kilogram of body weight. So this is effective for kind of restoring muscle mass if that's been lost, but also helping to really support with the amino acids coming in through the diet to ensure that we're getting as much as possible and as good quality as possible. And then I look for good quality carbohydrates. These can be useful. Gluten-free organic tends to be best. People tend to respond better to that. However, of note is that some people are really achieving some benefit with ketogenic diets. And this ties in really nicely because ketogenic diets are used to treat epilepsy. So there is a rationale to support the use of this diet. You know, low carbohydrate diets with or without intermittent fasting can be really beneficial for some. And again, this is just for some people. Not everyone is going to respond to this kind of intervention. It's all really highly personalized. And then I'm looking to introduce colour and diversity through fruits and vegetables, herbs and spices. This is going to introduce fibre to support the gut microbiome, which is what Dr. Galland has talked about. It's going to introduce natural sources of polyphenols, flavonoids, antioxidants, extra vitamins and minerals to support inflammation and redox reactions. And then I don't think any of us are going to disagree with the fact that processed foods, refined sugars, high fructose corn syrups, and highly inflammatory seed, corn, canola, vegetable oils are going to be a bad thing. The beneficial effects of removing these foods from the diet can actually come quite quickly because these foods contribute quite heavily to the reactive oxygen species and the activation of the immune system that can exacerbate certain symptoms. So I prefer to move people from seed, corn and vegetable oils to high quality olive oils. I might use fish oils and then I will, where appropriate, will be using fish in its natural form and animal fats. And again, this is where appropriate because not everybody handles dietary fats in the same way. So I'll be really careful about how I apply this in clinical practice. So all in all, dietary interventions and addressing gut health can be a powerful tool in reducing inflammation and IDO production. It's going to protect mitochondrial function. It's going to reduce the effects of reactive oxygen species, but it's also going to support the kineranine pathway into safely producing NAD+. And then with regards to lifestyle intervention, so I place focus on reducing stress. This again is going to support inflammation and reduce the expression of the TDO and the IDO enzymes. So I'll be looking at things like, encouraging people to take time out in nature, take time away from their screens, meditate, turning off social media and disengaging from Facebook groups and media outlets that induce or maintain a constant state of fight and flight responses. And then I'll look to reduce toxin exposure. This is going to take pressure off the gut, the liver, the kidney axis, and this can look like something really simple things like filtering water or removing plastics as much as possible from the environment, and then eating organic food, or at least reducing as much as possible pesticide and herbicide exposure. And then you can look at swapping things like cleaning products and personal hygiene products to less toxic alternatives. And another thing I really like to do is support the oral microbiome. So it's now widely accepted that oral hygiene has far-reaching effects on the body, and actually some microbes within the oral cavity can activate the immune system. So, what we're trying to do within all of this is kind of support the immune system and take the pressure off of it as much as possible so that we can reduce the expression of these inflammatory cytokines and the IDO enzymes. So I have some pretty robust oral hygiene protocols that I'll provide to some patients if they need it. And then lastly, I wanted to say to, all of those suffering with long COVID, and indeed anyone suffering with any unresolved chronic illness, is that, I know how debilitating and frightening this experience can be. But one thing I do say to all of my patients is to kind of stop researching and start taking action, because when you're looking at scary research papers and news articles, it's really not helping. It's actually impacting this pathway quite radically, and hopefully it's now clear that why we need to kind of step away from this and start taking action is going to really help promote this pathway into a less inflammatory way. So there are solutions. It might take a while to find the right one, but in my experience over the last 2 years, I find that the clients that really tune out of information outlets that promote negative thought patterns and behaviors have really improved a lot quicker. The recovery rates have been a lot more robust. So it has been an absolute pleasure presenting here at the 4th Long COVID Coalition, and thanks for having me. And also thank you to everyone who has been organising these events, and thank you for listening.
Thank you, Rochelle. That was great.
Thank you.
So I'll start with the question, Rochelle, and I'll hand it over to Dr. Garland. So I would just like to know what are your thoughts about the ketogenic diet, or if we talk about a kind of an intermittent fasting. So what are your thoughts on that, and how do you think it correlates with the pathway?
So I think it's highly individualized. So I tend to do quite a lot of testing on my clients when they come through. So I'll be seeing how they're handling fats. I'll be seeing how they're handling things like carbohydrates, because I don't see any point in taking carbohydrates strictly out of the diet, because ultimately that is our quickest source of using energy. So once I've really assessed the patient, and depending on how they can handle a ketogenic diet, then I may recommend it under supervision. But it's quite a radical thing to implement, and I find people who are really suffering with long COVID, it can be quite detrimental, because it will initiate that initial period of low energy, and some other nasty side effects. So, it's very, very personalized, and it all depends on the initial presentation of the client.
Sure. I would also just like to check with you, like if certain people been already prescribed with certain medications, what they are taking alongside for the treatment for their long COVID, or even if they have a COVID, and you do a personalized kind of a plan for them. Do you think it varies, and how do you manage those things, and what are the things you take in consideration?
Yeah,
to give?
so a lot of people are coming to me with polypharmacy. So that's a really big thing that I see within my clinic, because I think what the trends that I'm seeing is that people with long COVID have had kind of pre-existing conditions that have been unresolved, and then it kind of COVID-19 or the infection seems to exacerbate those symptoms even further. So yeah, I get a lot of polypharmacy, but I've got an excellent drug nutrient interaction checker, so I will not prescribe any supplement until I've gone through quite rigorous checks to make sure that there's no interactions going on whatsoever.
Right. Thank you, Rachel.
You're welcome.
I just want to say something about intermittent fasting.
Yeah.
There are definite beneficial effects on the gut bacteria that result from intermittent fasting, independent of the nature of the diet. I think the ketogenic diet is so unpredictable in the way that it will impact different people, and actually the way in which it impacts the gut microbiome of individuals. There are situations in which a ketogenic diet actually, in patients with multiple sclerosis, for example, helps to restore a healthier gut microbiome, but it takes 6 months to a year.
Mm-hmm.
The intermittent fasting was shown to increase the ability of clostridial bacteria to produce this neuroprotective anti-inflammatory metabolite, indole-3 propionate, IPA. And so there may be a specific benefit, at least in laboratory animals, that hasn't been looked at in humans, but there may be a specific effect, anti-inflammatory effect of intermittent fasting on the impact of the gut microbiome.
Right.
I think the other thing that I would add about the ketogenic diet as well is that if someone has got quite a lot of gram-negative bacteria within the gut, you can end up with translocation of endotoxins in the presence of a high-fat diet. So that's another thing that you have to take into consideration before adopting ketogenic diets.
Sure. And you was talking, Rachel, about the free radicals as well in this pathway. So do you think they are good or they are bad? But because it varies, and in different diseases, it don't consider as sometimes good as well. So what are your thoughts on that?
Yeah, so within this particular pathway, there's a lot of free radical production going on anyway when it's working optimally. And in response to those free radical productions, you're going to get antioxidants being produced as well within that pathway, and it balances itself out perfectly, because we do need a certain degree of reactive oxygen species to destroy bacteria and to support our immune function. But what happens when you're expressing this pathway too much, you're kind of overburdening that reactive oxygen species mechanism. So then you're going to start to override the capabilities of the antioxidant mechanisms in that particular pathway. But no, when kynurenine pathway works perfectly well, you know, the antioxidants free radical production, it balances itself out perfectly.
Sure. And sorry, Dr. Galland. Yeah.
Oh, yeah, I just had a question for you, Rachel, about supplements. Whether you have experience using tryptophan in patients with long COVID. Right. I don't either.
I don't do it. The reason I don't do it is because I feel that it displaces other amino acids. So, you know, we need the whole spectrum of protein, really. So I kind of focus on food.
And I've read that the majority of NAD plus in humans and other mammals is recycled. That is, it's the rescue pathway.
Yes.
Is the more active than the primary nutritional pathway.
Yeah.
Do you ever use niacin, NAD, or various forms of vitamin B3, nicotinamide mononucleotide or riboside? And what's been your experience?
Yeah, so I've used flushing niacin. I've had good results with flushing niacin. Again, you've got to be careful, you know, what you're doing with patients with regards to that. But again, with the B vitamins, I kind of try and apply as nature intended. So, you know, all of the Bs all together is what I prefer to do. Now, I have used, some of the other components as well, but I tend to, my approach is basically get the diet right first. If there's still nutritional deficiencies, then we'll go in with supplementation after. The thing that I find with these niacin type supplements is that you need other nutrients to kind of get them in and get them going. So sometimes it's not very good to kind of just go in with these high doses of one single nutrient. Yeah.
I was going to ask a question for the audience, so I'm just going to throw in this question before we move on. I'm wondering if a fecal transplant could be of help. He had autoimmune problems for 30 years. Any thoughts about that?
Quite a lot of animal studies that show that that's really useful. Dr. Galland would probably be able to answer that better than I would.
There's a lot of data on the potential benefits of fecal transplants. And I've seen some dramatic improvement in different individuals. The donor is the critical factor. Or actually, there are two critical factors. One is the prep that you give the recipient and to prepare the gut for the transplant. But the donor is really critical because I've seen people get much worse after fecal transplants as well.
Excellent. I think I'm going to have to move us on, but I'm sure there are lots more questions at the end, just to let the audience know that we'll be having an open forum after all our speakers have finished their presentation. So moving straight along, I'll let Dr. Kamal Dua introduce our next speaker.
Thank you, Philip. So let's move to our next speaker, Dr. Stephanie Seneff. Dr. Stephanie Seneff, author of The Toxic Legacy of Glyphosate. She's a senior lecturer fellow. She's a senior research fellow at MIT. For over 3 decades, her research interests have always been at the intersection of biology and computation, developing a computational model for human auditory system, understanding human language, applying natural language processing techniques to gene predictions. In the recent years, Dr. Seneff has focused her research interest back towards biology. She's concentrating mainly on the relationship between nutrition and health. Since 2011, she has published over 24 papers in various medical and health-related journals on the topics such as modern-day diseases like Alzheimer, autism, cardiovascular diseases, analysis and search of databases of drug side effects using NLP techniques, and the impact of nutritional deficiencies and environmental toxins on human health. Today's talk, what Stephanie is going to talk about is on long COVID, neurological aspects, role of senescence, and the treatment options. So without any delay, I would like to invite Dr. Senath.
Hello, everybody. Thank you for being here. Can you see my slides? Am I good?
Yes, it's absolutely great.
Great.
Thank you.
Okay, good. Thanks. Just wasn't sure. So long COVID, neurological aspects, the role of senescence and treatment options. And I want to say first, thank you for talking about my work on glyphosate, my book, Toxic Legacy. I noticed both of the speakers in passing mentioned organic food or glyphosate. I think it's a really important aspect of long COVID to make sure that you eat a certified organic diet. I think it may be one of the most important things you can do to protect yourself from long COVID and to treat long COVID. Stay away from the toxic chemicals in the food. You know, the water, the air, those are problems too, but the food is very, very important, and especially the glyphosate. I won't be talking at all about glyphosate in this presentation, however. So here's an outline of my talk. First, what is long COVID? Second, neurodegenerative disease. The third topic, very, very interesting to me, is syncytia and senescence. And then finally, natural treatment options and a summary. So people have sort of assumed that everybody knows what long COVID is, and I'm sure this audience probably does, but some information from the CDC, it can last for weeks or months or even years. It's found more often in people who had a severe case of COVID. The symptoms often resemble myalgic encephalomyelitis. It's a big word, myalgic encephalomyelitis, or chronic fatigue syndrome. And so some of the common symptoms include this post-exertional malaise, exhaustion, heart palpitations, headache, brain fog, lightheadedness, depression, anxiety, joint muscle pain, diarrhea, lots of problems. Fatigue being the most overwhelming problem that people face, fatigue and brain fog. And then here's another poster from the CDC showing approximately 1 in 5 adults, which was shocking to me. 20% of people who get COVID have long-term consequences over the age of 18. And these consequences are can cover all the many different aspects of the body, cardiovascular conditions, kidney failure, respiratory problems, musculoskeletal, blood clots, and of course, the neurological and mental aspect, which is what I'll be talking about today. So what causes long COVID? This is still, I think, more or less a mystery, although there's a lot of hints that it's coming out of the gut, and certainly from the previous two speakers. And I would agree with that. I think that's where it's being held. The virus is surviving in the gut, or just the proteins are surviving in the gut, not clear. But that's maintaining an inflammatory state in the gut. And then that causes mechanisms that cause the virus to spread. So this is where I'm going to go into this issue of syncytia, where the virus is able to spread from cell to cell through a novel mechanism involving syncytia, where multiple cells merge together into a giant cell. And then the spike protein is extremely interesting, acting as a prion-like protein. It can penetrate the brain barrier and it can induce neurodegenerative disease. So that's what's really scary about this virus. That protein is very toxic. And the virus actually induces a state of senescence in the immune cells or even in tissue cells, the neurons, the fibroblasts, et cetera, the heart, cardiovascular cells. So these can cause these cells to be sick. So just starting with neurodegenerative disease, can SARS-CoV-2 productively infect the upper or lower gastrointestinal tract? I've seen from my previous speakers that they have shown that this is very clear. And even whether it might be acting as a bacteriophage and infecting the bacteria in the gut. Very, very interesting question. But it seems that the virus can be sustained in the gut for a long time after the person has recovered, or at least the proteins that are produced by the virus are somehow being held by some certain cells and kept alive. And so those proteins are causing these responses. There's a high rate of fecal RNA shedding, and this was talked about by the other presenters. And viral RNA has been detected in these fecal samples. And so we need further study to figure out exactly what's going on in the gut. But it is clear that that's where it feels like the bacteria are helping, collaborating with the virus to keep it alive in the gut. So this is an interesting paper from 2021, Parkinson's disease and post-COVID-19 syndrome, the Parkinson's long COVID spectrum. And they looked at 27 patients who had Parkinson's disease and then caught COVID-19 across several centers. And they found lots of evidence of post-COVID-19 syndrome. 85% of these patients continued to suffer after they had recovered from the disease. And they had lots of worsening of the various functions that are associated with Parkinson's disease. The motor function, they needed more L-DOPA to maintain their ability to move. They had increased fatigue and cognitive disturbances. 22% had cognitive disturbances and sleep disturbances, but 85% had long COVID, and then you have half of them with these other problems. So clearly a bad effect of COVID-19 on Parkinson's patients. Now, this paper, I think, is very interesting. It's from 2013, so it has nothing to do with COVID-19, but it really paints the picture of what I think might be going on in the case of long COVID and how it can cause Parkinson-like symptoms in the brain. And so, first of all, there are terminals from the dorsal motor nucleus of the vagus nerve that sit within the gastric submucosa just a few millimeters away from the lumen. So very close contact between the gut and the vagus nerve. And now exosomes are a really important part of the process. These are lipid particles that are released by cells under stress, especially by the immune cells under stress, and they can contain the spike protein itself, because these immune cells that are getting infected or getting exposed to the virus, they pick up the spike protein and they release it in the form of exosomes. And those exosomes can then migrate to the brain and cause problems. They're delivering the spike protein to the brain. They also contain alpha-synuclein, which is upregulated in response to the virus. And then they can become toxic to neurons. This has been shown in studies that they're toxic to cultured neurons, the exosomes that are produced by the immune cells that are infected with the virus. So alpha-synuclein migrates from the gut and from the nose to specific regions of the central nervous system, and there they can cause then these problems. The earliest symptoms, when you're looking at Parkinson's disease patient, early on, before they've even expressed symptoms, they've got problems with the smell. The sense of smell starts to fade, and they're having, and the dorsal motor nucleus of the vagus nerve those are the early indicators where Parkinson's seems to first start, with this alpha-synuclein misfolding. And then aggravated alpha-synuclein from the affected neurons can be transferred to unaffected neurons via these exosomes. So the exosomes allow the immune cells to deliver the toxic alpha-synuclein along with misfolded foreign proteins such as the spike protein. Excuse me. And then they can transfer from the immune cells to the neurons and from the neurons to more neurons. So this is really a nasty consequence. Now, there's papers that are showing that SARS-CoV-2 proteins are, in fact, prion-like, and they can be the protein, the external protein that you're exposed to that can cause this whole process that I just talked about in the previous slide. SARS-CoV-2 proteins interact with alpha-synuclein, and they induce these Lewy bodies, which are the characteristic feature, these precipitates that contain the alpha-synuclein, the spike protein, the proteins in the virus can cause this to happen. And so alpha-synuclein is upregulated in cells that are exposed to the virus, and then it gets aggregated. It's accelerated aggregation as a consequence of both the S protein and the N protein, the nucleocapsid protein. And then they showed that these proteins cause Lewy-like pathology in the presence of excess alpha-synuclein. And then by confirming that SARS-CoV-2 proteins directly interact with alpha-synuclein, our study offers new insights into the mechanism underlying the development of Parkinson's disease on the background of COVID-19. And I should point out that this spike protein is quite interesting, has 7 of these classic glycine zipper motifs that are characteristic of prion proteins. And there are 7 of them in this protein, which is quite unusual. So now this slide, this is an interesting paper here, interactions between SARS-CoV-2 N protein and alpha-synuclein accelerate amyloid formation. So it's the same message again. The nucleocapsid protein speeds up the aggregation. Multi-protein complexes form amyloid beta plaque. And our results point toward direct interactions between the N protein of SARS-CoV-2 and alpha-synuclein as a molecular basis for the observed correlation between SARS-CoV-2 and Parkinsonism. So now my next topic is syncytia and senescence. Senescence-associated inflammatory responses, aging and cancer perspectives. Now, this is, again, a paper from pre-COVID paper 2015 talking about senescence, and it's a very interesting cellular response to injury. And it results in a permanent arrest of the cell cycle. So the cell sits there, but it doesn't die. This is the critical thing with senescence. And it sits there and it is metabolically active, and it produces these cytokines and chemokines, various kinds of chemicals. It's called senescence-associated secretory phenotype. This phenotype of the cell causes it to release metabolically. It's metabolically active and causes it to release a lot of proteins that exert a paracrine effect on other cells. So it's putting these out into the environment. The other cells are picking them up, and then they can also respond by becoming inflammatory and also becoming senescent. They link senescence to inflammation. These things that they release link it to inflammation. This is a very, very fascinating paper to me that I found only recently, a very new paper, 2021, micronucleus production, activation of DNA damage response, and C-gas sting signaling in syncytia induced by SARS-CoV-2 infection. And they basically looked at these HeLa cells that had been engineered to have ACE2 receptors. They don't normally have that. And they were grown in culture. And then they exposed them to the spike glycoprotein. And the ACE2 receptors allowed them to take it up. And they formed these multinucleated syncytia. And this is a schematic of it, two cells coming together, multiple cells ending up with a single giant cell with multiple nuclei, and also with these micronuclei, which are an indicator of DNA damage, DNA double-strand breaks. And that, of course, can induce serious problems like cancer. So the syncytia succumb to DNA damage in response to this, to infection with the virus. And that leads to genomic instability, causing these micronuclei to appear. And then the C-gas sting signaling pathway is a characteristic feature of this senescence-associated sensory phenotype. So this is another paper that talks about infection spread through cell-cell contact. And this can be because these cells blend their boundaries and are able to just push the protein right over without ever going into the extracellular space. So this sort of evades neutralizing antibodies because the cell's just giving it to the other cell directly. And so the syncytia formation is a way to do that. And all these cells come together, they all get sick with the virus. And they can also form these really fascinating, they send out these filopodia, which eventually can become tunneling nanotubes. And this cell can just deliver the virus and lots of other things as well to the recipient cell. SARS-CoV-2 infects cardiomyocytes and induces syncytia formation. This is a fabulous paper, and it has some beautiful figures in there. I show one of them here with these filopodia linking these two cardiomyocytes. So these are heart muscle cells that are blending together connecting with each other through these filopodia in response to the SARS-CoV-2 infection. And so, in fact, these muscle cells in the heart contain abundant levels of ACE2, so they very readily get infected by the virus if it gets into the heart. And then they noticed that the viral transcripts accounted for 88% of the total mRNA being produced by these infected cells. So they were just focused on making virus. And the particles ended up covering the cell surface of these cells. Fabulous pictures in this paper. And then the spike protein induced this filopodia formation, and then it caused fusing of the cardiomyocytes, generating these syncytia. And so furin cleavage, actually the furin cleavage site, which is a unique feature of SARS-CoV-2 compared to SARS-CoV, that was essential for the syncytia formation. You needed to cut the spike protein in half into S1 and S2. And then there were abundant progeny variants that were found in these exosomes that were released by these cells. So here's another paper. Post-acute COVID-19 is characterized by gut viral antigen persistence in inflammatory bowel diseases. And others in this conference have talked about this already, but quite fascinating. The RNA was expressed in the gut mucosa 7 months after acute COVID in 32 of 46 patients who had IBD. And the nucleocapsid protein was also detectable in 24 out of the 46 patients in the epithelium of the gut and also in the CD8 plus T cells, the immune cells. And then the antigen persistence was not obviously related to the severity of the original disease. But when they looked at the post-acute sequelae, the long COVID conditions, they were predominantly present in the patients who had persistence of the viral antigen. So it looks like the viral antigen is a causal contributor to long COVID, but they didn't prove that. So whether viral antigen persistence in and beyond the gut underlies the pathophysiology of post-acute COVID warrants further clinical trials. Here's a paper from 2022, the role of senescence in the chronic health consequences of COVID-19, innate immune system dysfunction that leads to decreased senescent cell removal or increased senescent cell formation could contribute to accumulation of senescent cells with both aging. That sort of senescence is a characteristic of aging and also with viral infections. And so it could be that the senescent cells, many are being created, or it could be the ones that are created not being removed by the immune system because the immune system is defective. Both of those are probably going on. And this is just a nice picture of the lungs infected with the virus, young versus old. So in the old lungs, you have a lot more immunosenescent cells showing up here. These guys down here, lots of different types of immunosenescent cells. They're surviving, but not, but just surviving and releasing chemicals that are going to cause trouble, inflammation. And so when you're old and you have a lot of those, you have a much more severe case, typically of COVID-19, and more inflammation because of the release of the cytokines and chemokines by the senescent cells. And virus-induced senescence is a driver and therapeutic target in COVID-19. So SARS-CoV-2 invokes a cellular senescence response in infected cells, which is accompanied by this senescence-associated secretory phenotype. It includes pro-inflammatory cytokines, extracellular matrix active factors, things that can break down the extracellular matrix, and also procoagulatory mediators that can cause blood clots. And then, interestingly enough, you can get secondary senescence of endothelial cells. It might not affect the virus, but they can pick up those secretions and end up also converting into senescent cells. And then the supernatant from those infected cells induce platelet activation and the clotting cascade. So again, those signals that are being released by those senescent cells are causing a lot of trouble. SARS-CoV-2 infection causes dopaminergic neuron senescence. So now we're getting into the nervous system. And this paper shows that infection in the neurons in the substantia nigra that are associated with defects are associated with Parkinson's disease. And when they're infected with SARS-CoV-2, an inflammatory response and the cellular senescence response are apparent. And these cells do express ACE2, and so that's going to promote viral proliferation inside these cells. And then they used a high throughput screen to identify several FDA-approved drugs that could help to rescue the cellular senescence phenotype. Riluzole, metformin, and imatinib were drugs that they found could be useful. And now, so this is really kind of a summary that brings together the problems with solving with the problems of the cellular senescence. I think that it all comes together in the long COVID neurological symptoms. You start with the misfolded proteins, and then their recognition as non-self proteins activates the innate immune system. And now that induces inflammation, inflammation induces cellular senescence, and cellular senescence induces more inflammation in a kind of a feedback loop. And then finally, the neurons get injured by this, and you get brain organ failure. Natural treatment options. Emerging senolytic agents derived from natural products. So there's several papers that talk about senolytics. I love that word because these are small molecules that can actively kill senescent cells. And this is what you need. You need to remove these cells. They shouldn't be alive. And so many things that we've heard about already, quercetin, that is a flavonoid in various vegetables, as well as in tea and red wine. Fisetin, which is a flavonoid in many fruits and vegetables, apples, grapes, persimmons, curcumin, which is a polyphenol in turmeric, and then piperlongumine, which is an active alkaloid found in peppers. So these are kind of the ones that you can eat and remove cells. So this is a paper specifically on the senolytic effects of quercetin. It means it can kill the senescent cells. Fat cells were exposed to reactive oxygen species, and they show that caused them to move towards a senescent state with these markers like senescence-associated beta-galactosidase activity, increased pro-inflammatory responses, and synthesis of MIR155, which is an inflammatory microRNA. And quercetin improved all of these markers. Many natural compounds have senolytic effects, and here's again many that have been mentioned already, phenols like curcumin and all of these things, organosulfur compounds, sulforaphane, which is found in the broccoli and all cruciferous vegetables, methyl tocols, tocotrienols, alkaloids like berberine, and piperlongumine, which was mentioned earlier, terpenoids like triptolide. So all of these good things are going to help to treat this treat these senescent cells. And then here's a paper on Bicalin showing that it actually helps to deal with prion protein misfolding, which is quite interesting. And so junk signaling is a key modulator in cell death induced by the reactive oxygen species. This Bicalin, it's the active ingredient in the dried root of a plant that's known as Huang Qin in traditional Chinese medicine. I think it's a very popular Chinese medicine treatment. It protects the neuronal cells against the prion protein-induced neurotoxicity by inhibiting this J and K activation. And it reduces prion protein-induced mitochondrial dysfunction and also reduces ROS. So this seems like a good option for treatment. And now I want to have a few slides here on melatonin. We'll go through them quickly, but melatonin highlighting its use as a potential treatment for SARS-CoV-2 infection. It looks like there's nothing not to like about melatonin. There's more than 140 scientific publications that have identified melatonin as a likely useful agent to treat COVID-19. It has a high safety profile over a wide range of doses. It doesn't have significant toxicity. It's a confirmed potent anti-inflammatory agent, and it's an antioxidant in many experimental models and clinical situations. And it's also a well-recognized suppressor of HIF-1 alpha, which is what is induced in response to hypoxia. And so here's another paper specifically addressing senescence. Melatonin suppresses senescence-derived mitochondrial dysfunction in these mesenchymal stem cells. So these senescent cells, of course, are generating reactive oxygen species, resulting in mitochondrial dysfunction. Melatonin can prevent the induction of senescence, and it enhances the therapeutic potential of these mesenchymal stem cells, help to keep them healthy. And it activates mitophagy so that you can clear the damaged mitochondria. So all of this is good news. And this is just a graph from a slide in a paper, another paper that talks about melatonin's many beneficial effects, and seeing all these different things that are suppressed by melatonin that are good things to suppress to try to tame the inflammation, to prevent the Warburg metabolism effect that's associated with cancer, where they're going into glycolysis instead of using their mitochondria to make the ATP. It can cause the M1 cells to mature into M2 cells, which are anti-inflammatory, and the protecting from the reactive oxygen species and toning down the inflammation. And so in summary, long COVID is a debilitating disease lasting for months. It could be due to persistent viral infection in the gut long after the acute phase. SARS-CoV-2 spike protein has prion-like properties, and this may be a factor in the neurological symptoms of COVID. SARS-CoV-2 infection is cell-cell fusion, leading to syncytial formation, and there's evidence of DNA damage and cellular senescence. And many natural molecules, including phenols, alkaloids, terpenoids, organic compounds, have synthetic properties. So that's good news because it's used to treat. And melatonin is a safe and inexpensive supplement exhibiting many beneficial effects, including antioxidant capabilities and clearance of damaged mitochondria. And thank you very much for your time.
Thank you. Thank you so much. Are you there, Dr. Seneff?
I am, yes.
Yes,
Can you
sorry.
hear me?
It was breaking in between. All good. Thank you so much. That was quite insightful. I would just like to know a little bit about melatonin first, because I can understand that melatonin can also be taken by quite a number of people for insomnia and stuff. Do you think it really nicely supports the tryptophan supplements?
Do I think it supports tryptophan supplements? Is that what you're asking?
Yep. And
it's derived from serotonin, which is derived from tryptophan. So in that sense, if you're supplementing melatonin and you don't have to make the melatonin, it may be helping to deal with the tryptophan deficiency problem.
Right.
I do worry about it because it does have such an important, such an important molecule in the blood indicating the wake-sleep cycle. So I wonder about if you're taking it in excessive amounts, are you disturbing what the pineal gland would normally do to control your wake-sleep cycle? I don't know. I know lots of people who are taking melatonin, even at surprisingly high doses. Personally, I would be a little concerned about that. And I don't know, some of you may have experience with that as well.
Sure. And what do you estimate about the possibility of developing these severe neurological disorders?
About the what?
About developing the severe neurological disorders.
What about it?
What do you think with the severity, how often it will develop with the long COVID symptoms?
How often, how much, those kinds of things. It looks like quite a few people are developing, brain fog and cognitive problems. So Parkinson's, of course, is a big one. And I believe that the virus is going to increase the the prevalence of Parkinson's, and we'll have to wait and see because that takes time to develop. But it looks to me like it's kind of a good setup for Parkinson's disease. So that's the one I would most worry about. But also the prion disease itself, which is very, very rare. But I think all of those, and Alzheimer's, dementia, I think all of those are problematic. ALS, because they're all connected to misfolded proteins and the prion-like aspects of the spike protein, and the fact that it can get into the brain are all very disturbing through these exosomes. I think that, to me, is a very worrisome aspect. And I think that if people have a healthy immune system, they can, of course, keep those senescent cells at bay. They can clear those senescent cells with a healthy immune system. But many people do not have a healthy immune system, and I attribute that in part to glyphosate. That's something that I, wrote about. I have a whole chapter on how glyphosate disrupts the immune system in my book. And of course, there are other chemicals that we're exposed to as well. So I think it's really important to maintain a healthy immune system. And I think one way to do that also is to get out in the sunlight. I'm a big fan of sunlight exposure because it's free, it's readily available, and it increases your vitamin D, helps to strengthen your immune system.
Yeah, and you have beautifully shown quite a number of nutraceuticals compounds like quercetin and all. The same question which I have asked previous speakers as well. Do you recommend any particular concentration which you reckon would be the best for those kind of compounds for the long COVID, or
I can't do that. I don't treat people. I have no clue, actually. So that would have to defer to the experts on that.
No, absolutely. Thank you. Yes. Thanks, Dr. Senath. Yeah, Dr. Chetty.
Stephanie, just to reiterate what you've just said, it's the infection with SARS-CoV-2 that will cause the syncytia formation, and drive cells towards senescence. And those senescent cells, which should be eliminated by the body, produce a whole lot of toxic chemicals which drive inflammation.
That's right.
That's one process that we're looking at. And then the second is with the spike protein acting as a prion-like protein on its own and causing damage, neuronal damage, as well driving alpha-synuclein production and causing aggregation, and in that way causing neuronal damage, as well causing the spread via exosomes, worsening that nerve damage. Mm-hmm. So those are the two mechanisms that we have at play. You mentioned the persistence of antigen, viral antigen in the gut itself. Is that due to bacteriophage activity that spike protein persists? Why do we have this increase in antigen for this length of time in the gut itself?
A great question.
Has they shown persistent viral infection in the gut?
Right, I know. Those are big questions, and I don't have answers. I think it's all very interesting hypotheses, and you probably know better than I do exactly. Well, you probably have a stronger arguments for your hypotheses, but it looks to me like I think there's a possibility that you mentioned, it was mentioned in an earlier talk about bacteriophage like capability of the SARS-CoV-2 virus, that it can infect the bacteria, and the bacteria can acquire DNA, coming from a viral infection. So it's conceivable to me that the bacteria themselves may have gotten some parts of the proteins in the virus incorporated into their own DNA, and now they're just producing those proteins. It's not a viral infection. It's just that the bacteria are expressing the proteins that came from the virus, segments of the spike protein, for example, that could be causing all this irritation and all these problems. It's possible. So I don't know. I think either continued infection hiding in the gut, or somehow some cells able to hang on to the spike. I don't know. I think it has to be some kind of continual production of the proteins of the virus, either by these bacteria that have acquired the genes, or by continued infection of the virus, hiding in, for example, senescent cells and sharing it, keeping it going. I don't know.
Can I ask a question on behalf of the audience? I thought this was an interesting one. What are the chances that long COVID is an autoimmune disease?
Yeah, I mean, that's certainly a possibility, because the spike protein, for example, has many, many segments in it that are similar to segments in human cells, human proteins that are produced by human cells. And so there are people who have shown that there's many, many different autoimmune diseases associated with certain specific proteins that have some sequences that are similar. So if you have antibodies to the spike protein, the whole thing could be driven by the antibodies without even having a continued infection. And that is a very interesting idea, that because of the overactive antibody response to the virus, that's now sustained and is now attacking human tissues through molecular mimicry. That's quite a possibility. I think that we should consider that. It may not be any continued infection. It might just be antibodies. Excellent question.
Great. I think we're going to move on to our next speaker. So thank you. And just you, I'm sure you'll be staying with us, Stephanie, for some more questions afterwards.
I will. I'll stay the whole time. This is great, by the way. I'm really enjoying these talks. They've been fabulous.
Thanks, Dr. Sanif. All right, so we'll move to our next speaker, Dr. Abdul Manan Beg. Dr. Beg is a medical doctor and a scientist from Pakistan. He has served as a physician in internal medicine at the University Hospital in Pakistan. He earned his PhD from the University of Sunderland, and after the outbreak of COVID-19 in early 2020, he has published over 25 papers on COVID-19 and SARS-CoV-2. His papers on COVID-19 and SARS-CoV-2 focused on clinical presentation, treatment modalities, and targeting vital targets in SARS-CoV-2 caught attention even in the international news outlets and researchers worldwide. Recently, he has published on neurological defects in the long COVID in Nature as well. Dr. Beg is a recipient of the National Research Productivity Award from Pakistan Council of Science and Technology and the Award of Excellence in Research from Aga Khan University. So Dr. Beg is going to talk about neuro-COVID, actions needed for prevention and treatment of neural damage. So let's welcome Dr. Beg.
Have you got, have we lost Abdul Manan? Can you hear us? What I'll ask you to do, Manan, is jump out and jump back in, but don't do yours. Let me control your slide because it may be the signal is too strong. I'll show your slides.
Can you hear me now?
Yes,
I'm
yes,
hearing you now.
absolutely. Yes, yes. Dr.
Okay,
Beg.
great.
That's great. Thank you.
So if you just put on my slides, okay, I can then continue with my topic, which is
okay. So
prevention
I will have control of your slides, and you just tell me when to move it on. Okay.
If you can, if I can change it, okay, would that be okay? Because it has got some animations.
Oh, I see. It has disappeared. It's okay. The animations will still work. You just have to tell me when to go ahead with it. So I've got it right here. I think we're still having a connection problem there. So can I ask you to just jump out and jump back in? Sometimes the connection resets when you do that. While we're waiting for Dr. Beg to come back in, yes, we have some great, thank you very much for all our audience. Thank you very much, Inka. We've got somebody who is a fan of Dr. Beg. Are you back with us now?
Yeah, I am here. Can you hear me?
I'm hearing you again. So we'll put
Okay,
your
good.
slide back on and we'll see. Go ahead.
Okay. So this slide shows you the progression of a disease which was initially thought to be a respiratory disease. And it shows that how you acquire the infection to how it goes to the brain. And in the slide, okay, you can see from the region of the nose,
Manan, the signal is gone again. What I'll ask you to do is just jump out of the studio and come straight back in. I'm sure the signal, yeah, wonderful. So yes, so whilst we're waiting on Dr. Manan Beg, I'll look and see if there are any quick points. We've got a few points here, shining bright light on the whole thing. Yes, thank you, Andy. And we've got Equilibrium. Thank you as well for sharing your thoughts with us. Mitch, thank you very much for being with us. We appreciate your presence. And is that better now, Manan? Can you go again?
Yeah, I can hear you. Can you hear me now?
Excellent. I'm hearing
Okay.
you fine. So you go ahead and I'll move your slides.
Okay, thank you very much. So this slide shows you that in the phase of acute COVID, one can have neurological symptoms as shown in diagram B. And it could be attack of the virus on A. Once the virus is inside the cell, it can cause development of certain deposits, okay, that you can see in the diagram B3, Philip. They include deposition of plaques like beta amyloid and even tau protein. So there is a question that is asked that if it goes beyond 3 months or years, okay, is it possible to diagnose that the neuroinflammation is still going on? On the spot B4, you can see I've shown some markers, okay.
We've just lost you again, Manan. I tell you
So,
what you do, Manan. Switch off your video.
okay.
Switch off
Yeah.
your video and let's see if you just use audio.
Okay.
Excellent.
So
Yeah.
the connection might be better now if you can hear me. What I was saying is that the CSF can have some elevated biomarkers as shown at a diagram spotted as B4. The question is that is the direct viral injury only explaining the neurological symptoms in long COVID? So it might be the to the brain, okay, caused by thromboembolic phenomena. If Philip could spot it on C, you can see that embolic phenomena and thrombotic phenomena can also give rise to hypoxia. Hypoxia can cause a neuronal damage. So the question is that with neuro COVID, what the person is I mean like is exhibiting in form of sign and symptoms that you can see now. Philip is showing you that there is a brain fog. I personally don't like this term because in detail where we'll talk about the treatment modalities. Next slide, please. This shows you the key players, okay? Persistent neuroinflammation, presence of SARS-CoV-2 and its associated protein. My earlier speakers, my peers, actually spotted the role of S protein and N protein of SARS-CoV-2. You can, at the upper right-hand corner, see the vascular event ongoing that you just heard can cause ischemia, hypoxia, and cause neurological deficits. Though it's
We just lost your signal again, Manan. You were just breaking up just a while ago. Can you hear me? Can you hear me, Manan? Okay, I think we're temporarily losing Manan here. So what I think we'll do, because this is a very important talk that Manan is doing at the moment with regards to neuro COVID. I'll try again and see if we can get him back in. What we may do if we are struggling is that we may shift into the open session and then come back to this if we can get the signal back again. Manan, is that clearer?
Yeah, now it's clearer, please.
Okay, go ahead.
So if you could move to the next slide, I could show you how the damages are taking place. Now, if you see, okay, the infection goes through cribriform plate, and it can infect the olfactory bulb as shown in the upper right corner. But there are places, okay, where the virus hideouts. You can see the and reside and can come back and then reinfect the newer cells. So these hideouts are important because the virus can emerge from here and cause a reinfection of the naive cells in the body. The reason many long COVID patients are having problem with sometime feeling better, at other time feeling worse, like that. Next slide, please. So here is shown that what sign and symptoms could occur in neuro-COVID. You can see them being listed from number 1 to number 2.
I think we've lost you again, Manan. And so I'll just keep talking through your slides here. Just as he said, neuro-COVID, memory loss, gait ataxia, convulsions, delirium, meaning confusion, loss of cognitive function, cranial nerve deficits, and loss of reasoning ability. So these are all, I guess, a number of things that Dr. Baig has been looking at with regards to the neuro-COVID effects. I think it probably is important to note that I guess these don't necessarily happen suddenly. These may be quite progressive over time, which makes it even more difficult, I suspect, for us to clarify if it's related to COVID-19 or something else. Are you back with us, Manan?
Yeah, I am here. Okay.
Yes.
So this slide actually shows you that what type of sign and symptoms the patient can suffer in acute COVID and carry it over to long COVID. And in the next slide, if you move on, you would see the evidence. Okay, many people ask that where is the evidence? Show that whether it goes inside the neuron and infects it. Okay, so at the regions of the star, you can see below in the black and white diagram, the inset is showing you the presence of SARS-CoV-2, not the RNA of it, the entire virus inside a neuron. So this paper was published and it came out in for COVID-19, but then just imagine that the same type of infection the person can be carrying into long COVID. Next slide, please. Now, this slide, okay, when I just made it, okay, I saw my earlier speaker went into quite a detail of it. You can see not only the virus itself, but the spike protein itself can cause formation of fibrils, which then subsequently become the beta amyloid and tau protein. At the top where the mouse is moving, okay, you can see spike protein can blend up with the amyloid fibril, and in the presence of a neutrophil elastase, which is a very crucial player, okay, can make super fibers, or these super fibrils, okay, get deposited in the neurons and they assume the form of beta amyloid and tau protein. So once these substances get accumulated, just imagine how quick the neuron would age. So a person in 30s and 40s would be like having neurons, okay, which are laden with these deposits, okay, which are reminiscent of Alzheimer's disease. You heard Stephanie saying Parkinsonism, and with Lewy body and stuff, okay, they would resemble many other degenerative diseases. The $1,000,000 question: what this virus sets up into the neurons? Okay, it sets up apoptosis, as you can see in the diagram in the below left, where the red arrow is coming out of the skull of the human, and it also causes the loss of brain mass, okay, which was shown for many patients with COVID-19. Now, the next diagram will talk about the role of glia and other stuff. But before that, I want you to see this, that there are several molecules, okay, which can target the pathways and the virus and the S protein as well. You can see the name of the molecules highlighted in blue and their chemical structure. They are very well known inhibitors of cytokines that are listed in green. There are certain drugs like piperine, okay, and hesperidin, rutin, which with the sign of inhibition are shown that what process they can inhibit. So I was just wondering that instead of these drugs alone, okay, if there would be a formulation which have these components in some nutritionally allowed quantities, remarkable effects could be observed. A newer player into the game is known as immunomodulatory drugs, which is shown on the top right with the name of IMIDs. Okay, one example of the drug is polymidomide and thalidomide family. It also has got a linedomide family. These are neuroprotective as they inhibit the alpha TNF, but then I came across very few of them from the literature that are as neuroprotective as the nutraceuticals that you can see on the slide. Like hesperidin inhibits the SARS-CoV-2 main protease as well as the spike protein. Rutin, you can see, is anti-apoptotic. It's doing the trick here by inhibiting the microglia-induced apoptosis. On the extreme right, you can see two major players, okay, the piperin and Michelin, okay, doing the inhibition of the cytokine. So if we manage to not only target the virus but also inhibit the inflammatory process, that could be a success in treating long COVID with neurological symptoms. Next slide, please. Now, dysautonomia is thought to be autonomic nervous system disorder, but then you see the surname says nervous system disorder, so it also comes as a subclause of neuro-COVID. Many patients, okay, countless of them, okay, are in long COVID are suffering autonomic disturbances. You can ask me a question, okay, how does the autonomic symptoms arise in patients with long COVID? The next slide will show you where actually the virus can target. Just look at the mid-sagittal section of the brain. You can see vasomotor center there written. Vasomotor center is an area which controls the autonomic nervous system. There are two very high players, okay, in the hypothalamus. The highest control showed as green and yellow. And just imagine that if these neurons get infected with SARS-CoV-2 or their spike protein, they are going to just like bring an earthquake to the autonomic nervous system, regulation of autonomic functions. The reason patients suffer not only tachycardia when they stand up from a lying position with POTS and they have got anatomical and physiological instability with their management of their blood pressure, number one, their heart rate, their even venodilatations. All of them are kept in check by the autonomic nervous system, which seems to be a very clear and evident target of SARS-CoV-2 in long COVID. Next slide, please. Now, this one is showing you that don't mix this amyloid with the cerebral amyloid that I was explaining, but in the periphery, okay, with chronic inflammation, there could be formation of amyloid depositions, which if occlude a coronary artery can lead to a heart attack. But our topic today, neuro-COVID, if you go to the top, you see it can even occlude a cerebral blood vessel leading to hypoxia. Now, don't mix up this one with stroke. This one actually partially blocks the blood vessel, so the blood flow to the brain gets limited, and then the neurons can suffer hypoxic damage that you heard Stephanie and other speakers were stressing that it can cause a neuronal atrophy and deposition of beta amyloid and tau protein. Next slide, please. Now, the treatment approaches. So what can be done? I mean, if you ask me a magic pill, okay, that would be a pill that has got anti-inflammatory effects, neuroprotective effect, could be antiviral at the same time, very preferably a nutraceutical, if not stem cell therapy, okay, which I'll talk in the next slide to show you. If these drugs and even the stem cell therapy could go into a clinical trial to see that whether it could improve the symptoms with neuro-COVID or not, it would be some practical thing to do. Next slide will show you that how the long COVID or neuro-COVID symptoms can be addressed. Number one, you should be targeting the viral loads. Number one. Number two, you should be targeting the re-entry mechanism, which is so very well known by the virus when it comes out from the hideouts, and of course the mother of them all, the viral replication. So how it could be done? Now on Twitter and several other social media channels, okay, very commonly you see posts, okay, especially when people ask that how viral loads can be eliminated. Simple cleansing of the nose. Okay, no need to use hypertonic salines. Simple inhalations, they can do the trick. Okay, if you do it daily, it reduces the viral load. I want the audience to understand one thing, that viral loads are not constant. Once you get an infection and it's not going out after 3 months or a year, that means your body incites of viral loads, be it colon or gut or nose or sinuses. It's continuously like the virus is coming out of the cells after infecting them in a very large quantity. So the viral loads may be building up and you might not be knowing it. You only suffer symptoms and you think that it's some cytokine thing. The cytokine is driven itself by reappearance of the virus which comes out of the hideouts. Re-entry mechanism is my targeting it is my favorite. I'll tell you why, because I've been experimenting with drugs and some in vitro results that if you block the S protein of and flip, if you can just click, okay, those molecules would appear. You just press the space bar. Yeah, you see, it's an animation slide. Now you see the drugs coming and blocking the S protein of the SARS-CoV-2. So if they block the S protein, it cannot sit over the ACE2 receptor. It's an inconvenient word, but I call it the castration of SARS-CoV-2 because it would not be able to do what it actually is destined to do. So it can forget about sitting over that ACE2 receptor, so it's as good as a dust particle. So there are drugs which can actually block this S protein of the SARS-CoV-2. You heard with you saw in the previous slide with hesperidin and piperin and rutin. They all successfully sit over this S protein and block it. If you see the third one on the extreme right hand side, you can see two nutraceuticals, okay, shown to block the main protease of SARS-CoV-2 at a site which is shown here. If you can click the space bar again, Philip, it would appear here at the socket. See, these two drugs, okay, have shown consistently to come bind at this site, okay, which has the inhibition site of the main protease of SARS-CoV-2. Next slide, please. So now, neural stem cell therapy, okay, seems to be very attractive, and many people with long COVID, okay, keep on asking the question, do we stand a chance with this? Now, there is something which I want you to know. If you could click the space bar, there is a problem with this therapy, and that problem would appear in the next slide. If you move on, Philip, thank you. You see, on the left-hand side, you can see areas where the neuronal loss are if they are there, okay, are very accessible to the hands of the transplant surgeon. And very usually it could come out as an uncomplicated surgery if the region is like very handy to approach. But if you see on the right-hand side of the slide, in a mid-sagittal section, which shows the presence of the neuronal damage or replacement that has to be done in hypothalamus or thalamus or the lowermost black circle, the medulla oblongata. These are very critical location. And let me tell you something very candidly, that the operative procedure to attempt to transplant cells here itself could cost a life, because you can't reach these places so very easily. It's very easy to say that you can see in the mid-sagittal section. Just imagine how a surgeon would approach these areas. Not possible. So if you click the next slide, okay, you will see that this is a critical location, and post-operative complications are there. So question is that can medicines and some regenerative what you call as growth factors can do that? Next slide, please. So you see, I'm not saying it out of clouds, okay? There are evidence-based literature, and you can see them from top to bottom, that the compounds Veclin and hesperidin and quercetin, they have been known for stimulating neuronal stem cells, and they actually touch those critical pathways, okay, which can cause differentiation of cells into neural stem cells. So my thing is that in cases, okay, as you saw in the previous slide, when the transplant surgeon is actually thinking of attempting those areas where even going there, okay, the operative procedure could kill,
These therapies, okay, would be more the mainstay of the neuronal regeneration in critical location type of transplant attempts, because then you won't actually wait for the post-operative complications to occur, because those are given in form of drugs. So if it's surface, maybe stem cell can stand a chance, but if they are deep inside the brain, forget about the procedure. I mean, like excuse me, some of them say that injecting the stem cell into the CSF can do the trick. Not at all possible, because it can go to other places and grow there and result in a dysgerminoma or a germ cell tumor. Okay, so transplanting at the accurate site is needed. If not, the next slide, please. These medicines, okay, for which I've shown you evidence-based papers here, one of them in scientific report Nature, okay, clearly indicate the role of curcumin and other molecules which can cause a neuronal regeneration. As you heard me earlier, okay, my thought is that if these chemicals and molecules and nutraceuticals could be given in a proper combination, they could serve much better than the agents alone. So next slide, please. These are the molecules, and you just see where they get published. I mean, like, it's not I mean, like just a proposal or something. Okay, this is a Springer Link journal where they showed molecules which are drawn and with their name, okay, to be anti-inflammatory, neuroprotective in their action. There are multi-potential compounds shown on the right-hand side. You heard me on them on previous slides, okay, where their anti-inflammatory and anti-oxidant properties were discussed. Some of them are brilliantly neuroprotective. The reason these would stand a new chance in addressing the neurological deficits in long COVID patients. Next slide, please. Brings us to the point. Who is going to do it? I mean, like, from where to get an advice? I mean, online telemedicine is going on nowadays for COVID. Even for long COVID, okay, there are institutes where you can consult for your neurological symptoms, as shown on the left-hand side. Always brilliant to ask patient, okay, in a consultation that what are the current drugs you are upon. Right-hand side of the slide, please. And if these two pictures are known, I mean, neurological symptoms can give an idea to a consultant that where exactly the focus is. And if the current drugs he or she is taking is addressing viral loads, re-entry mechanism, and viral replication, very good. But if they are not targeting like any of these or all of these, then I say that there is a chance that neurological symptoms would reappear, or there is a chance of a relapse. The question, how would we know that which patient with long COVID needs what therapy? You just see at the arrow pointing below, individualized therapy. That means not every person would need the same amount of anti-inflammatory and neuroprotective drugs or a neuronal stem cell therapy. You have to individualize the patient according to his or her needs. So, but then at the end of the day, the surname of the treatment is anti-inflammatory. It should be neuroprotective. It should have a antiviral component, and if accessible to surgical hands, okay, why not even a neural cell stem therapy? So with this thought, there was a proposal, I mean, like, to how to address these things. Okay, what should the patient prepare? What should be I telling to my consultant when I visit him? Next slide, please, the last one. This one tells you that, first of all, your online telemedicine consultant should be the one which by reputation knows. If he knows, okay, book a consultation, get a Zoom session or a Skype or a team, whatever you like, the arrows which are going below. So treatment options, okay, discuss with them, and almost always request that he should brief your local consultant, who may be sometime reluctant, okay, to talk to a doctor from online telemedicine, but then just ask him that like he has got some thoughts, okay, why not just listen to them? To the patient, I would say keep it handy. Number one, your current lab results. You see in the yellow, your current lab results should be with you. Your current drugs that you are taking should be with you. The list of them. You should not be searching while while having a consultation, get them prepared, and be very prepared to ask question. I mean, just the one below it, yeah, and don't hesitate. I mean, even if you want to ask that, I've got dysautonomia, is the virus in the hypothalamus or medulla oblongata, or is causing demyelination of my nerves? What chance do I stand, okay, with the multivitamins, nutraceuticals, or even multivitamin injections with the myelin sheath regenerate or not? Can my neuron come back or not? Like, feel free. I mean, this is your time. If you don't know what you are going for, it's very, I mean, consultation into clouds. So the patient should be ready with few things. Your online telemedicine consultant should talk to your consultant in real life. Okay, write him a letter or what. You can ask me a question. Okay, like how would a online telemedicine guy would examine doing a neuro COVID telemedicine session? You see 3 slides on the extreme right hand side, please. We can test cranial nerves. We can check for various signs like rigidity, tremor. You can save the patient to perform certain task, okay, which gives clue towards the neurological symptoms. So it's not totally impossible to have a neurological examination online. But if you go and your consultant knows, okay, what you should do, okay, in order to give him clue that which part of your nervous system is possibly affected, I think it would be a brilliant thing to do. At the bottom, you can see COVID-19 written with a link. This is one of those 75 sites that I know that perform consultation for COVID-19, acute phase of COVID-19. But we need to follow them, okay, and develop sites like these, okay, where telemedicine for long COVID should also be available. I mean, like, I mostly do it on Twitter, okay, but then there should be many country-based, globe-wide online sessions in order to do this. Thank you very much.
Thank you, Dr. Baig. That was great. So I got a couple of questions, and then I'll let Jim ask you some few more questions. So, Dr. Baig, I just want to know, you have highlighted quite a number of nutraceuticals in your slides, and that's quite interesting because my lab also significantly focused on those nutraceuticals, and I have clearly shown that they have potent anti-inflammatory and even anti-cancer activity in various lung diseases, apart from lung cancer, asthma, and COPD. Okay. So what do you think, what are the pathways you are looking here that in these neurological disorders, these nutraceuticals are targeting?
Now, that's the beauty of an alkaloid or a neuroceutical particle, that when you actually develop it from scratches, okay, you know a single pathway or 2 pathways that a drug could target. These compounds for centuries are known for their effects in infections, especially viral infections. Some of them were even used as Chinese medicine, okay, for influenza and other viruses, after which we noticed Parkinsonism in 1918. So you name it. I mean, with the compound that you saw, they are antioxidant. At the same time, they are neuroprotective. Over 53 papers on them. And I keep downloading those PDF, okay, and to just like reconfirm what else it could do, But the charm is in their combination. I mean, that's one thing I would like to emphasize, please.
So is there any particular molecular or cellular target you think it's a common among all?
All of them, okay. If you are talking about neuro-COVID, the answer would be different. If you're talking about long COVID, okay, then the pathways would actually disseminate. Okay, I mean, so right. If you say or neurological or neuro-COVID, they are anti-apoptotic. They prevent the development of amyloid. Some of them have been reported to clear the amyloid, okay, and tau protein. So I think that for neuro-COVID, that's the answer. For long right. COVID, the pathways are like, you won't believe, okay. I know guys, okay, who have got more than 250 pathways, okay, on a single wall, okay, with these molecules acting,
Yeah. So in continuation to the same question, do you think the brain of the long COVID patients would exhibit amyloid, tau, or any other deposits?
Now, this is a question that I will answer now, Kamal.
And we will come back to this, okay, via either email or a similar talk in 23 or 24. The answer is yes. Okay, yes in caps. Ask me why I am so sure about it. Okay, I'll tell you why. See, if a person had acute COVID, okay, and then after a year he's having similar symptoms, you tell me what actually he's carrying. A malaria? I mean, like, I don't understand. The same virus is there for some reason or the other. You can't detect it, or it has shedded proteins which are there, which are causing the problem. So the question is that in acute COVID, you have found beta amyloid, tau protein, okay, and you name it, okay. So if it's a low grade process, you will see. I wish that my patients with long COVID live enough and get treated, never get autopsied, you know. But the few cases that we will have, okay, in 2030, 2022 and 24, Kamal, we will have a session, okay, in which I will tell you that in 2022 I told you.
Sure. And what do you think with the brain fog? You the term which you don't like. What would be the basis at a molecular level for that condition, like what you call brain fog?
Okay, Kamal, I'll put an example so that you and the audience would understand it very quickly. Okay, so if I ask you, okay, there are two goats, okay, hunted by a lion, okay, and the lion actually would do what to them? Okay, so how much time would you take to say that he would catch one of them and the other would run away? Okay, believe me, even this simple question, sometimes the guys would actually take time to compute to understand that what he's asking. This is brain fog. Okay, they look at a zebra crossing on the street. Okay, it looks like a tunnel vision. I mean, like, should I walk or should I stop? I mean, like, memory recalls. Okay, cognition problems on the top of them, 7 to 8 of them. Where is the fog part of it? Okay, I don't see fog. You see in a rainy weather, you know. So this term came, it grabbed, okay, and it's there, but it should not be misunderstood. All the brain fog complaining patients have got severe cognitive dysfunctions.
Sure. No, thank you. Thanks, Dr. Big. I'll hand over to actually Yochim. Yochim, you are on mute.
Hi, Manan. Yes, after we have spent several weeks and I don't know how many hundreds of papers we have exchanged on all this, let me emphasize one thing maybe that was not really well highlighted. We're looking at these fibrils forming, especially from the Swedish study, where you could see that the spike protein dropped in serum and watched under an electron microscope would then dissolve itself into amyloid beta fibrils. And especially if you add neutrophil elastase to that, they would become cleaved and become shorter and much more toxic. And my suspicion would be that in that, and similarly the question, will these fibers then persist and will they go and form thrombosis, and can they form amyloid or other aggregates in the brain at the same time? Are we looking at the same main source for these two very prevalent conditions in long COVID?
Yeah. It was on one of my slides at a 12 o'clock position, okay, where I showed spike protein dissolving in amyloid fibrils and forming superfibrils. See, when I say superfibril, okay, just try to understand it, that this is something the body has never seen. $1 million question, can the body dissolve it? Okay, $1 billion answer, it cannot. Why? Because it has never seen that. So then comes the point what to do with it. Okay, you deposit it, and then it comes into traces. Okay, whenever you do the autopsy, that actually is the fate. If it dissolves in a blood vessel, what would you do? You can't remove it. I mean, like, because you have never seen it. So if you have got a product, okay, for which the body is very new, okay, to it, okay, from where it would bring the enzyme to dissolve it. The reason the patients are the clots are difficult to actually dissolve, the reason these amyloids are being tagged by ubiquitination proteins, but they are not being cleared by proteasome or endosome mechanism. Joshim, answer to your question is, it's such a new thing, you don't have an enzyme for that. You take 1,000 years or more than that to evolve to form an enzyme for it.
But one more comment to that. We are like we've been discussing that almost for a year now, that we're looking at multi-component fibers, and that is now the proof for that. So if you have a multi-component fiber consisting of a spike protein of amyloid plaque or amyloid fibers or beta and neutrophil elastase, then taking away the building material of that, like stopping spike from spreading, calming down the neutrophils and bringing down the allostase, and also reducing amyloid beta, which we can do with the molecules. So we do have a chance of cutting that cycle, at least prevent it from forming.
Very right, very right. And you know what those compounds that you saw in chemical structures do? They actually remove the amyloid fibrils. Okay, so tell me where the spike protein will dissolve with. When it comes to prevention of formation, yes, it is very much possible. But I'm alerting my listeners. Okay, once formed those superfibrils, you can't dissolve it. It's very hard because we don't know what it is. So if you treat them with the chemicals that I showed you on different slides, the amyloid fibril would clear. It would go bind the spike protein. So you don't allow the ingredients to mix and make that superfibril thing. That's, Josham, very much possible.
Yeah, but one more comment to that. We had the same, where we couldn't observe it better than in the brain is, of course, in the blood. And so we had these results that were done on a patient where you could see that a triple anticoagulant would not do much to these thrombi because of that reason. Because they don't work on these mechanisms. And then we had them take some nutraceuticals only for 3 weeks, and they would have a insignificant reduction in these blood clottings. So I think there is hope. And we can measure it actually in the blood, not in the brain, because that's so difficult.
Very, very difficult, because you cannot take a biopsy from a living guy, So I mean, dissolving them could be an issue, but first thing is that do not allow them to get formed in the first place.
Yeah. Okay, but that we can cover, and that looks pretty good.
Yeah, why not?
Excellent. Thank you very much, Aman, and thank you very much to our panelists. I'm going to give Valentina a quick chance to update us on some information that we think is important. So I'll hand it over to her now. Do you want me to share your slides?
Not at the moment, so you want to.
Okay, wonderful. Go ahead, Valentina.
So, distinguished speakers, panelists, and viewers, it's my great honor to be a co-organizer of this 4th Long COVID Coalition Congress on Neuro-COVID. And I am very grateful for the knowledge that you all have shared today. I also would like to express my highest respect to Joachim Gerlach and highlight his fascinating, independent leading research work, which led us to look into Neuro-COVID in a more structured way and cover in depth many pathways which drive the anxiety, depression, and neurological issues like cognitive dysfunction, brain fog, seizures, and many other symptoms in long COVID. I should also highlight that the presentations from all speakers were built on a joint collaborative research effort of more than 1,500 research resources in 2 weeks' time. Therefore, I would like to congratulate all of you for making this collaboration a real success. We have focused on Neuro-COVID this time because we wanted to raise an alarm. Lack of treatments or lately provided treatments can increase the prevalence of Alzheimer's disease, dementia, or other neurological chronic diseases. The future will be built by us, all of us here today, and those who watch us, those who treat long COVID and research it. But in order for us to succeed, we need to address 2 conditions. One, we, as doctors and scientists, need to act together for communal benefit, so that we care not only for benefiting individually, we act for the benefit of all who need us, for our communities. And 2, we need to collaborate. We need to engage with stakeholders, doctors, and scientists more widely to make progress on tackling this disease with right measures. So I would encourage those who work on long COVID, who makes life-changing research, please check our website, which is still in development, but please get in touch via email. So details of Long COVID Coalition website are now on the screen. So please try to email if you do work on long COVID. We want to hear from you, and we want to jointly solve these unsolvable problems for people. So And Philip, if you could share some slides. Thank you. So today I'm honored to present Dr. Carlo Bronia findings from our second Long COVID Coalition Congress and his treatment protocol, which in some ways has evolved with time. So this is just another example on how Long COVID Coalition brings change in understanding the disease and helps finding the appropriate compounds to tackle the mechanisms, not symptoms. Next slide, please. So where we are now, there are 2 million people with long COVID in UK alone, and around 180 million people worldwide. There is no approved treatment for long COVID 2 years from the beginning of pandemic. And I want to raise question to anyone in a position to answer. Why, with such huge in-depth research that we are showing every Congress, we still don't have approved treatments? What we see is happening is there are empirically validated treatments that have been shown to be effective in treatment of people with PTSD, anxiety, and cognitive impairment. This was according to psychologist James Jackson, who is director of behavioral health and Vanderbilt University Medical Center ICU Recovery Center. In one of the interviews, he said that while new approaches to treating long COVID patients may emerge, many existing psychological and behavioral tools, such as acceptance and commitment therapy, cognitive behavioral therapy, group therapy, and peer support, for example, seem to be effective treatments for different aspects of long COVID. Then another similar comment was made by neuropsychologist Dr. Renee Matahill. Patients struggling with insomnia may benefit from CBT, while those reporting brain fog may need to work with cognitive rehabilitation therapists and discover compensatory strategies. So instead of understanding why people develop these symptoms, there are such comments made as potential treatments for neuro-COVID. We think this is wrong, and I would encourage patients to be more vigilant when they're offered such treatments for and after COVID developed depression, insomnia, anxiety, or other neurological symptoms. Next slide, please. So healthcare systems so far failed to provide adequate treatments for one or another reason, but we care and we want to find solutions for the mechanisms which were discussed today. Next slide, please. So going back to our second Congress, Dr. Carlo Bronia presented his lab findings, where he clearly shows that SARS-CoV-2 has a dual mechanism. It infects human cells, but first it infects bacterial cells in our microbiome. As a consequence, there is release of toxins that go into bloodstream, disturb the CNS and gut-brain axis. And I would encourage all doctors who treat long COVID patients, look into these images. The virus is within bacterial cell, and it is there. It is on the images that have been taken for everyone who treats long COVID. So next slide, please. A very important step in Carlo's treatment is to stop toxins as a first step. And for adults, main thing is to consider a combination of amoxicillin, clavulanic acid, probiotics, and medicinals 9. This combination will tackle bacteriophage behavior, stop any viral proliferation in the gut lining and bacteria replace, and control bacteria that got killed by SARS-CoV-2, will help restore gut microbiome, restore dampen epithelial cell inflammation, restore mucus layer, restore tight junction integrity, and finally maintain gut homeostasis. So for adults, amoxicillin clavulanic acid for 2 days, 3 times a day, then twice a day for 4 days. For children, amoxicillin and clavulanic acid are prescribed according to body weight for 6 days. there are alternative options if you're allergic to amoxicillin. Next slide, please. So in long COVID, we have very similar combination of these solutions. And the important is oral therapy. So as our speakers also have mentioned this step is very important to clear toxins from nose and throat. And we again see the amoxicillin or rifaximin together with probiotics and bisphenols 9. So I should highlight that for the probiotics, as per Dr. Leo Galland, Bacillus subtilis has also shown to be very effective and can be used as an alternative to Lactobacillus plantarum or Bacillus clausii, and again, used temporarily for 15 days at least. So in terms of medicinals, for those who is not aware what it is, this is a solution which passed 3 stages of clinical trials. It is unique pharmaceutical-based therapeutic suspension consisting of natural compounds like bakelin, quercetin, curcumin, EGCG, luteolin, hesperidin, and more. And I should say I was very pleased to identify this solution a while ago, and I was very pleased to be able to record an interview of clinical trial data, and feel free to check Long COVID Foundation's YouTube channel if you're interested. So for children, we also applied the same tactic, and we tackle bacteriophage behavior with amoxicillin and clavulanic acid according to body weight for 6 days. So that's all from me. And once again, I would like to thank everyone who participated today and for such joint effort to address so many pathways of neuro-COVID, which have been overlooked before. So if anyone from the panel would like to share clinical experience based on amoxicillin and flavonoids combination, please do so. Otherwise, I pass the stage to Philip. Thank you.
Thank you very much, Valentina. Really appreciate it. Very important stuff. Listen, this is the part here. This is a very long session, but extremely important. And we really appreciate our listeners who have stayed with us for this duration. We're almost at the 3-hour mark. But I think that what we're going to find is that the work that has been done through the coalition, especially highlighting what Valentina and Joachim has been doing at pulling these speakers together, this is going to form a resource that is going to train people, I think, for years to come. It is so far ahead in terms of the thinking and the research. And so it will take some time for people to catch on to just how significant this is, but I believe that it will happen at some time. So I think this is a time for us to just talk for a little bit before we would close out our session. And I've had the pleasure of listening to everybody. And can I ask a question? So one of the questions I want to ask is long COVID random, or does it affect specific people? Now, this is an extremely important question to me because a lot of people think it's just random. If it's just random, we cannot predict who may be at risk. We cannot do anything preventative. We cannot do anything, so to speak, in case they get infected. So from my perspective, from a clinical point of view, as with many other diseases, we identify risk factors, we manage risk, we reduce the risk of long COVID. Does anyone have any thoughts about what are the characteristics of people who may be at risk?
Well,
oh, no, you were okay, Leo. You weren't muted.
okay. There are a number of studies that have attempted to answer that question. Here's the big problem with that. I think we know a lot about the pathophysiology that leads to long COVID. I think that there are elements in that pathophysiology that can be the target for therapeutic interventions at the time of acute COVID or before. And I have a protocol that I call long COVID prevention that I use with my patients based on that. I've seen people who were really, people who come in to consult me, they were really healthy, they had great diets, they were athletes, they had no illness, and COVID left them, and they didn't even necessarily have severe COVID. COVID left them just devastated. And sometimes it wasn't immediately after COVID. They seemed to recover, and then something else happened, and then they crashed. So even though statistically we can identify who's at greatest risk, people who are overweight, women over the age of 50, people with a history of asthma, those with low levels of IgG3 or IgM. These have all been reported in different studies. There are people who have none of those risk factors, who really have their lives transformed by this condition. My view on this pathophysiology is that depletion of ACE2 and mitochondrial damage are key factors from which a lot of the other pathologic events emerge. And so what I do in treating patients with acute COVID is I try to follow their symptoms very closely. And if I don't see that, I use treatments that have as their goal enhancing ACE2 activity and supporting mitochondrial function. And I use that at the time when they're ill. I would say that in my practice, the incidence of long COVID is somewhere less than 2%, not anything like the statistics that are routinely reported. So that's my perspective on prevention. I want to say one thing about the amyloid and about the unfolding protein, the misfolded protein response that Stephanie talked about, which I think is so important. That may be happening primarily in the brain. And that is scary because how do you get in there? There is evidence from Parkinson's disease that it could start in the gut. And what's encouraging about that is maybe we can really get in there and do something about that much more easily. This research from Rush University, and there have been several other centers, but specifically the work from Rush University in Chicago, where they looked at people with early Parkinson's disease and they looked at gastrointestinal function. And most people with Parkinson's disease have GI symptoms before they develop Parkinson's disease. What they found was inflammation in the colon with increased colonic permeability, not anywhere else in the gut, just in the large intestine, and the formation at sites of inflammation in the colon of alpha-synuclein. That it appeared the alpha-synuclein was traveling from the colon into the central nervous system along the track of the vagus nerve and maybe other enteric afferents to the brain. So I do think that, I was, a year and a half ago, I was pretty skeptical that the gut was going to be a key battleground for COVID-19 or long COVID, even though my background is in dealing with the microbiome. But the evidence that keeps accumulating has led me to believe that I should have seen that.
Can I say one thing on that before? Sure. The thing that stood out to me was the research paper showing that 40% of patients who had inflammatory bowel disease ended up with long COVID. That to me indicates a very serious risk factor. The other one that I would look out for is interferon autoantibodies.
Right, that's definitely associated with mortality and severe acute COVID. I had seen a paper which found that there was a negative correlation between spike protein antibodies and the development of autoantibodies. That somehow, so the theory was that the autoantibody formation was interfering with neutralizing antibodies being formed. I do think that autoantibodies play a major role. Are they occurring because of the T cell dysfunction, or is there some other driver? And just to take this further, and I think this is an area that really needs to be explored. Two things about antibody synthesis in the course of COVID-19. And I'm sorry that this is so complicated. There's a study from the University of Arkansas that found that the majority of people with acute COVID had developed autoantibodies against ACE2 that persisted after recovery. These were functional. So this might explain the ACE2 damage that occurs in people who do not, who aren't very sick to begin with. But they found that these autoantibodies, the timing of these was very much related to the development of spike protein antibodies. And what they postulated is that there is something called an immune network that forms, that is the antibodies to the spike protein are generating antibodies against the antibodies. They're called idiotypic antibodies. And then there are antibodies generated towards those called anti-idiotypic antibodies. And that the anti-idiotypic antibodies were actually the autoantibodies to ACE2. So there's a lot of complexity. I've approached immunologists about, well, what can we do about that? I haven't gotten any answers. I have tried proteolytic enzymes as a way of trying to break up these antigen antibody complexes that are forming, which is what these immune networks are. One other study that I think is really important. It was a study that looked at one of the mysteries of the early part of the pandemic was that people would get sick. They would seem to not be so sick for the first week, and then suddenly at about day 7 or 8, they would just crash and develop a cytokine storm. And there was a South African physician actually who claims that by giving people steroids on day 8, he saved everybody. So there is this fascinating study that was published about a month ago. ACE2 is not the only cellular receptor for SARS-CoV-2. There are other receptors that have been identified. Monocytes are white blood cells that do not have ACE2. Yet Patterson in his work found spike protein in senescent monocytes. He thought it's because they were gobbling up the endothelial cells where the virus was. It turns out that there is a protein on the surface of the spike of monocytes, atypical monocytes, called CD16. And that is a receptor for the spike protein. The virus can enter cells that express CD16 on their surface, and aged monocytes, non-classical monocytes, senescent monocytes express CD16 on their surface. They do that under the influence of various inflammatory chemicals, in particular something called fractalkine, which is associated with coronary artery disease. It may be one of the reasons why people who have coronary artery disease are at greater risk for severe outcomes, because they have more of these monocytes. Once the protein enters the monocyte, it induces a state of pyroptosis. That is, basically the monocyte bursts into flame, in essence, microscopically, and initiates the cytokine storm. The timing of that corresponded to the development of antibodies to the spike protein, suggesting a phenomenon that had been feared and which has been described in things like dengue fever or Zika virus, called antibody-dependent enhancement. Interestingly, this only happened with natural antibodies. It didn't happen with vaccine-induced antibodies, which is, I mean, that's what most of us were afraid of, that the vaccines would induce this. In fact, the natural infection was able to do this. So, I mean, there's something in the antibody response to this virus that is part of why people are getting so sick. And it's very likely that it may be part of the long COVID phenomenon. I view, and I'm sorry for going on and on like this. I can get kind of worked up about this, and I haven't come up with all the answers, but I do think that balancing and getting the proper T-cell immunity is really going to be the solution, the long-term solution.
Yeah, go ahead, George. You're muted at the moment.
Sorry about that. Yeah, thank you, Leo. I think there's one thing that was in the slides for the preparation of this conference, which I included, but we didn't find that anywhere with the speakers, that the T lymphocytes are being directly infected. And there's even a picture under the electron microscope where you can see the T lymphocytes filled up with SARS-CoV-2 virons, as we could also see with the bacteria. But maybe for us, to Philip's question, what could be the underlying factor of why people do get long COVID? That is a question Winston Churchill once said when he was listening to all his generals. He said, like, oh, now I have the sum of all fears. So if you go just onto our pathway list that we have, which is now around more than 200 single pathways, which are kind of self-perpetuating and fueling each other, and in an in an awesome interplay. That sometimes when we are looking at all these studies over the last few months, it was like we were concluding, what is this? I mean, Stephanie has gone through the glyphosate molecule for, I think like around 8 years, and has published, I don't know how many different pathways to disease. That's even the name of your paper. And I'm so grateful that you've been my teacher before COVID came, because that is exactly the point. It's a fantastic construct, this spike protein and this virus by itself. I don't know how that came about, but I'm in awe as to the sophistication of what it can cause in self-perpetuating and self-fueling destructive cycles. Compared to that, glyphosate is almost a nutraceutical, as I have to say. So the question, who's going to get long COVID or not? I mean, if you have a dysbiosis and you're preconditioned, it's like an old building. If an earthquake hits, then a building that is unstable will crumble first. I mean, that's in the nature of the thing. But by which way you will crumble, there is hundreds of ways how this COVID virus can get to you and what it can trigger, and it depends where it comes in. And like I said, it can infect your T lymphocytes directly. It can infect your cardiac pacemaker cells. It can go into your brain. It can go anywhere. It just depends a little bit. Plus all the variants, you were talking about host cell receptors. We have been trying to cover, and I think we do successfully cover, more than 12 different host cell receptors, apart from ACE2. So that was in the early research. We covered everything from CD147 to DPP4 and all the other ones known that are from other coronaviruses or previous coronaviruses like MERS, SARS-1, and also the so-called flu coronaviruses. Plus covering another, I think 12 or 14 different enzymes, cleavage enzymes that the spike protein or the virus needs for fusion or endocytosis, etc. So that is all possible, but you have to have early patient treatment. So what we are looking at here is the damage already being done, a patient not being treated early on, and then having these symptoms. So in my opinion, it is very much possible that a very healthy person gets infected with SARS-CoV-2 and it just hits his T lymphocytes, and then he gets immunodeficient in a way, and then he upregulates bacterial co-infection, gets dysbiosis, and you just name it. So it can come in through every crack, and it will break open every crack it finds. It's a very detrimental molecule.
Go ahead, Abdul. You're muted.
We actually come across this question in our long COVID Congress and Foundation each time we debate any other topic or a discipline. The answer to this possibly lies in the type of the immune response a person actually mounts. And we are genetically programmed to mount a particular type of immune response to a fixed dose of viral load, you know. So if we mount a moderate response to clear the virus, okay, we won't go to long COVID. If the response was subtle, it partially cleared it, virus remains, goes to hideouts, comes back again, okay, residual virus, residual protein, okay, long COVID. So a suggestion was that, and I published on this one, and somebody like universities in in UK and US are following that line, that it's the HLA antigen of what we are made up of that determines the type of the immune response we will mount. If we completely eradicate it, okay, no question of long COVID. I mean, like, even it could do it in the gut, it could do it in the sinuses, it could do it in the tonsils, everywhere. If you are unable to completely eradicate it, then it remains okay, and then it shows off. So that HLA someday, possibly, in my opinion, would answer the question that a particular subset of types of HLA antigen could possibly predispose towards long COVID. My take on it.
Yeah, I'll tell you what, and this is my challenge always. I say categorically, if we do not figure out exactly the pathophysiology in long COVID, we will go in a circle. We have to find the fundamental pathophysiology. And here's another thing that I strongly believe. There is nothing that is new. Everything we see has happened before. The challenge is to find the parallel disease. So far, and I think I'm getting closer to it, the parallel disease to long COVID is Epstein-Barr. Epstein-Barr, however, does B cells. It seems as though SARS-CoV-2 does T cells. But it is going to be a parallel disease, and we have to figure out. The problem we've got is we haven't properly figured out Epstein-Barr either. Long Epstein-Barr. People have it for years after infectious mononucleosis, and we manage them as chronic fatigue and whatever else. So the challenge that exists for us today is that in order to solve long COVID, we have to be willing to solve all those diseases that we have ignored for the past 20 or 30 years.
I agree. Yeah, you're right.
May I say one more thing? Today we have, the conference was actually split in two different topics. One was the first part, looking more at the, more like the chemical disbalances, like the neurotransmitters and the NAD plus and mitochondrial mechanisms. And the second part was actually addressing something else that is not really playing out yet, not in large numbers. And so apart from curing long COVID, actually the idea of us to have the second part of that conference is as a warning. Now, like Stephanie has been warning for 10 years about what glyphosate will do in Alzheimer's, autoimmune disease, and autism, and you just name it. This SARS-CoV-2 will be worse. And yes, Epstein-Barr will contribute, and other pathogenic overgrowths as well. And we could name another 50 that play a role and that are all upregulated, like Lyme, like fungal diseases, like just normal bacterial co-infections, biofilms that are settling because of pathogenic overgrowth, and that will be the breeding ground for amyloid formation, and so on. It's, like I said, it's an intricate, awesome mechanism that we are facing here. And what we want to say, without creating any panic, look, what can we do when once these are manifested diseases? If you have Parkinson, if you have multiple sclerosis, if you have dementia and Alzheimer's, it's going to be much harder to do anything about it. So why we do this is to say, look, let's look at that and find more proof, and let us see how we can prevent it from happening. And maybe the people that are facing long COVID right now are more prone to long-term neurodegenerative disease than people that have no problems right now, which doesn't exclude that they might get some trouble with prions or whatever is in the forming. And that is the idea.
Yeah. Can I ask Stephanie a question? There's something that you said today that I had only come across recently that really worried me. I didn't know a lot about exosomes.
Yes.
Now, what is technically the difference between an exosome and a lipid nanoparticle? Are they not technically the same thing? It's just that all cells produce exosomes. So it's such an important question in my mind, because if any part of the virus or potentially even the vaccine, we still have to explain long vaccine, could that then become packaged in an exosome and shared?
That's what I think is happening. I think that exosomes, so you're right, they are lipid particles. They're small particles. You have LDL particles that are also lipid particles, and they're very different. But the exosomes, it's increasingly becoming clear to a lot of researchers that the exosomes are maybe the primary communication network among the cells. And when a cell is stressed, it basically dumps its load into the exosomes and ships them out. And it's partly because it's not able to turn it into a lysosome and digest it and break it down. So it has these toxic exposures that it can't clear. And it's in the endosome. And then the endosome is like, my gosh, what am I going to do with this stuff? I can't clear it. So it says, okay, I'll just pitch it, just throw it out there and let somebody else deal with it. I think it's a way that for a particular cell to sort of temporarily solve its problem by just giving it to somebody else. And then these exosomes travel very nicely along nerve fibers. And this is the whole thing with Parkinson's disease. I knew about this before COVID because I've studied Parkinson's pretty heavily. And I was aware that it starts in the gut, and you have these immune cells that take up these, prion-like cells that are produced by pathogens, and they carry it into the spleen. They take it into and then they release these exosomes both from the gut, the gut's immune, the gut's immune system, and also from the spleen. They release it into the circulation, and it travels along the nerve fibers. And those nerve fibers make a very beeline for the brain, and you go right into the brain centers, even the ones that control breathing and heart rate and consciousness. Those can get hit hard. I think we're finding people, I suspect, there's issues with even just getting the parts of the brain that are really critical for main function of the body to keep it alive can get inflamed and to the point of even killing the patient. So I think it's really scary to think about these exosomes and their ability to travel throughout the body along the nerve fibers.
I'll tell you what, is that I'm going to make a prediction here, and I'm trying not to be too dramatic with the prediction, but there's something that has been floating in my head recently. It is that I suspect that we're going to see a lot more spontaneous brain bleeds because of that amyloid. It's what we call cerebral amyloid angiopathy. And it's a predisposition for hemorrhagic strokes. If what we are saying is accurate, what we should start to see an uptick of is that we're seeing an increased frequency of hemorrhagic spontaneous strokes. And if that's the case, that is a very big worry and something. This is part of the reason why we need to get a handle on this very quickly, and we need to identify who is at risk. This is why I'm emphasizing this so much. Go ahead, Joachim.
Great. If you want to do something quickly now, we were talking about these three components that would lead to these clottings and to this thrombi and so on. So the neutrophil, the control of the neutrophil activation is the key point. Bring down neutrophil elastase, and then you have taken out one of the major components that can fuel this process.
Yes, I understand. But I think even so, I guess part of the reason I'm asking these questions is because so much of medicine is about predictive work, meaning that we know that if you have hypertension, you're at risk of heart disease. So we therefore manage your hypertension 5 to 10 years before to reduce your risk of heart disease. We don't want to treat the fact that you've got a heart attack. That's what we used to do. And so medicine always requires a degree of predictability as to risk. And this is what I'm saying is that we're missing at the moment. We need to know the risks. We need to preempt the patients who are at risk. We need to be able to intervene easily years before this happens. And so the big part of the challenge is, how are we going to do that?
Listen to Rachel and Leo, and do everything you can in your food, in your diet. Avoid toxins, eat well, and sleep well, and drink clean water, and eat good food, organic. Stay away from glyphosate, etc. Bring all the possible, how you say, the chances in your favor and not in your disfavor to start with. That's the easiest. Doesn't cost much.
And I think we already have 200 million people globally who need treatment. So we already have a lot of people to treat. And I think it's best to identify the solutions and apply these solutions already and prevent this mass of people growing further. I think that's my point. And I would like also ask Dr. Shankara Chetty, maybe you can share your experience on tackling COVID acute.
Yeah, Valentina, I think that with the numerous mechanisms at play, treatment of long COVID is going to have to be individualized. We know about the immune dysregulation that occurs a week after the start of the acute illness, and that seemed to have a genetic predisposition. We know about the infection of the gut microbiome, so that'll play into it, and there's a diversity of different gut microbiomes in different individuals, which will increase their propensity of developing infection in those bacteria. There's also the toxin production in the gut itself, with neurotoxins, conotoxins, and the like, that need to be addressed as well. So I think each patient will have to be examined individually, and we'll have to try and find out the different mechanisms at play for individual patients and individualize their therapy. But I think the conferences that we've been holding are vitally important in understanding the different mechanisms at play. And so we can examine patients and figure out exactly what's at play with each individual patient and figure out a treatment strategy for them. And I think that's what's most important.
Thank you. I think we're coming close to wrap up, but I wanted to ask Rachel a question. And this is something that I've been wondering about. When we talk about fecal microbiome changes in long COVID, what's very interesting is that you have fecal microbiome changes in celiac disease, in fibromyalgia, in chronic fatigue syndrome. Are the fecal microbiome changes that we're seeing in long COVID present already, or is it because of the infection?
And I remember we've spoken about this before, haven't we? And the problem is when I get clients coming to me, they are already in the long COVID phase. So when I'm doing gut microbiome testing, I'm only getting a snapshot of what's happening at that particular moment in time. So I don't have any kind of pre-issues. But there is one specific client that I did have that did actually come to me years ago for small intestinal bacterial overgrowth. And we kind of treated her and she went off on her merry way. And then she had a really perfect kind of microbiome, so to speak. But she came back to me with long COVID. And so I've got the comparison of the kind of I've got 3 tests actually for her. So it was really interesting to see that she had low butyrate producing bacteria that had contributed to her SIBO, and she had low lactobacillus and she had low bifidobacterium. So she didn't have anything specifically pathogenic in her gut. She had a tiny little bit of Clostridia, but nothing too horrendous. And then we got her microbiome back up to kind of what is a normal microbiome, but we got her symptom free. And then when she came back into clinic, those exact same markers had come back down. So is that due to COVID-19? I don't know. But that's the only correlation that I've got. That's the only one person I have with pre kind of COVID gut microbiome. So from my perspective, when I'm doing these tests on my clients, there's not really anything, there's not really any significant gram-negative bacteria going on. There's no significant destruction to the gut wall, so to speak. It's mainly these commensals that are being kind of targeted, and these butyrate-producing bacteria. That's the major correlation I can see in my clinic at the moment.
I'd like to speak up on that because in my book, I talked about glyphosate in the gut. That's a chapter in my book on glyphosate. And I showed that glyphosate has been shown to cause an increase in the pH of the gut, of the colon. The pH becomes too high. And with a low pH is what you need to make both butyrate and acetate. So those short chain fatty acids, the microbes that make those short chain fatty acids become depleted because the environment is too basic. There's not enough acid in the environment because of the glyphosate. A very nice study showed both a correlation between the glyphosate levels in the gut in these mice and the levels of acetate production and the pH. So all of that worked perfectly with statistically strongly significant correlations between glyphosate levels and pH and acetate production. And butyrate also needs a low pH in order, those microbes need a low pH. So I think there's a critical aspect here of glyphosate exposure causing disruption of the microbes that make those short chain fatty acids.
Yeah, I agree completely with you. And, even here in the UK, a lot of the kind of nasty pesticides have been banned, but there's still residue around everywhere, and we're still using toxic herbicides and pesticides. And I think, glyphosates are a really big problem. I completely agree.
Listen, I think this was a phenomenal discussion. I really appreciate all the speakers. And I have to say a very big thank you to Kamal. What time is it there now in Australia, Kamal?
It's 7:17.
So he has been with us
You
since
can
about
see the sun is just coming out. It's
It's
really a winter morning.
So we really, really appreciate that, Kamal. That has been a
Well, it's
tremendous
a pleasure. It's indeed a pleasure for me listening to such an excellent speakers and learning more about neuro and COVID and long COVID. This is really a great platform, and it was great talks. Really great.
Thank you very much. And again, I say a special thank you to Joachim and Valentina, part of the Long COVID Coalition, who has got these speakers together. Long may your work continue. It will be appreciated many years down the line, finding all these people to focus their research attention on long COVID. And I'm sure the patients appreciate it. So any final words from Valentina or Joachim as we close out?
Maybe just short. Thank you everybody for all your incredible effort and taking the time and your expertise. And I hope that we meet again in a month's time for hopefully a little bit more positive topics. Yeah, thank you so much for participating and in the name of the Long COVID Coalition, all the patients and everybody in the industry. Great job. Thank you so much.
Valentina? Yeah.
Thank you very much. And without all of you, these congresses wouldn't be real success. So the knowledge that you bring, the clinical expertise that you bring only fuels our interest into identifying critical causes of long COVID. So thank you for participating. And just maybe only one thing is to, there are some patients who try to ask their own medical doctors to apply one or another treatment based on all this knowledge that is available. And for some reason, doctors dismiss them and they say that I don't have time to read this new research. I've got our own stuff to do. So you as doctors, could you please advise what is the best approach to help patients to get to these doctors and maybe somehow collaboratively generate something that majority of long COVID doctors would look into this research and properly treat patients, not dismiss that we discuss for 4 weeks now, and we still have lots of information to discuss in the future, but we need your advice as doctors as to how we can more widely to communicate that information to those who treat long COVID. What patients can do to support that?
This is a big question, Valentina, and here is I'm going to be honest with you with the answer. In reality, long COVID is a reflection of what we have done with fibromyalgia, chronic fatigue syndrome, post-Lyme disease, and Epstein-Barr virus. And the reality is that medicine has turned its back on it because it didn't figure it out. What we have here is a situation where the patients have got to insist. It's going to be a fight, because the history is that if we don't understand it, we walk away. And that's the truth. If the doctors don't have a clear protocol in their mind, a clear understanding of the science, they will walk away. And this is why I've been so focused on trying to find primary pathophysiology, because if it can't be explained to them in a way that makes sense, where the treatments are reasonable and safe, they will not do it. That's a huge challenge. Yes, go ahead, Abdul.
I managed to talk to 7 or 8 of those physicians, and the patients, while visiting them, made a direct call with his consent of the physician that if you would like to have a few words. I mean, like, not everybody accepts that. Okay, some guys have ego issues, but those 7 to whom I talked, they actually were very, eager to listen to what I was saying about them, that the patient is suffering, and some of them nodded. I mean, like, I was amazed. I mean, like, they would hear, but then, as you said, you have to insist. If you don't, okay, they do their casual job, okay, and then they walk away.
We know a lot about the pathophysiology of long COVID. I mean, there's still major gaps, but what we need to start developing training programs for physicians.
Yeah, absolutely.
Based on what is known.
Yeah. Wonderful. Thank you all. Go ahead.
Leo, just to add on that, in Australia in particularly, there are long COVID clinics now specifically, and they are specifically managing those patients, and they're maintaining in such a way so that if something work on certain patients and a similar case can be observed in future, those things can be applied to that. So most of these states in Australia now, they are having a specific long COVID clinics.
That's brilliant.
That's wonderful. Wonderful. Again, thank you all very much. Wonderful to have you all. Tremendous. I absolutely love these, and we learned so much. So thank you for our audience for staying with us, and look out for our next coalition conference, hopefully coming up in about a month's time. Have a great evening.
Thank you so much. This is wonderful.
Thank you to everyone.
Nice weekend. Thank you.